Good morning, Doc, and all the members of the platform.
Good morning. How are you?
Please, I'm fine.
Okay, we can great.
And you
It's
too.
past 7, but it seems uh the rain and then the chilling weather is still keeping people on bed. So we have to wait a little bit. So welcome. I can see Anita. I can see Anita there. I can see Praise. Oh, my Lord, the Kingsley, you are there. Belinda is here. Gloria and King Bashiru. King Bashiru. I hope all
Hello.
Yeah, I hope everybody is doing great.
You're doing great, Doc.
Uh,
Thank you. And how are you doing?
I'm doing all by God's grace. Thank you. The grace is keeping all of us. Thank you.
Good morning,
So
Doc. How are you?
I'm doing well, Madam Anita. I'm fine. Thank you. We should have to wait a little bit for folks to join. Okay. I think what we are left, it's not all that much. Is photosynthesis is respiration and photosynthesis basically. So I will take my time and then walk you through this one. But even though it's not that much, I wouldn't rush it to do all of it today. The reason being that I mean, it's it's not difficult, but it involves a number, especially the photosynthesis part. It involves a
Yeah.
lot of reactions, and then I would love that you really understand the concept very well. So today I intend to just do the respiration and fermentation, and then we get one more day, and then do the photosynthesis so that you get a firm grasp of everything. Especially once I know that you'll be teaching in SS, and some of you will also be going for your masters and PhD. So which means that it is very important you understand the concepts right. Okay. So all right. So I think Cecilia has just joined us. Okay. Victor, you are there. Okay. Victor, how are you doing? Okay.
I'm doing
King
well,
Victor.
and you?
Yeah, that's great. Good to see you. All
Great to
right.
see you too.
That's great. And then where is let me see, Cecilia, how are you? Sissy, how are you doing? Cecilia, you just joined. I hope everything is fine. Okay. Yeah, it's Sissy. Okay. Let me see who is here again. All right. Okay. So we'll wait for a couple of people to join and then we'll kick start. Okay. But it's nice to see you here. Okay. Great. All right. So we'll wait for a couple of people to join and then we'll kick start. Oh, okay. Anna is here. Anna, welcome.
Thank you, sis.
Yes, I hope you and the family are doing very well.
Very, very well.
Oh, well, we bless God. Okay. Okay. Oh, my lady, the Salamatu has come. Okay. All right.
Good morning, sir.
Salamatu. Good morning. How are you?
Fine, thank you, sir. How are you
Oh,
too?
I'm doing well by God's grace. Oh, Rosamond, how are you doing?
Doc,
Rosamond?
please. Doc, please, I'm fine.
Oh, we bless God. Thank God. Oh, my one gifty is here. Gifty, are you there? Welcome. Gifty, are you there? Yeah, man. Yeah, man. Yeah, man. Yeah, man. Gifty has given me some back. That's beautiful. Yeah. So we wait for a couple of people. We are just 15 here. I don't know why. Maybe because of the weather. Okay, folks are finding it very difficult to get up from bed. So wait for a couple of minutes and then we'll kick start. Okay. Thank you. I hope you are going through the trial questions that I sent. please make time to go through them. Okay, the one that I sent. Make time to go through them. Okay, if possible, answer them. You can WhatsApp them to me. I look into it for you. Okay, and then Philomena, how are you doing? Philomena, are you there?
Yes,
Hello, how are
please.
you? Oh, that's great.
Mm.
Right now, the King Ajungo has joined. King of Tema.
Oh,
Okay. Ajungo,
So yeah,
Charlie,
yes,
what's
sir.
up? Everything cool?
Bro, Ade, good morning, sir.
Good morning.
Yes, I find by the
I
grace
hope
of God.
it's. Oh, you can't go. All right. So
Yeah, you can't complain. Yes, sir.
we, we are waiting for few colleagues to join and then we'll take start. Okay. All right.
Yes, I've been to other centers. I was just doing my survey round, and it seems we are the, for want of a better word, I say we are far behind them. I think
Oh,
West
yeah.
Coast discussed photosynthesis last week.
Yeah. Okay. No problem. You don't have any problem. Okay. We want to, well, the most important thing is that I want to go in a slow pace so that you understand everything. Okay. So don't worry.
And
You,
I think
you,
this also
you'll be fine.
as well.
Okay. You'll be
As well.
fine. I mean, it's better.
I know because everything is coming from here. So I'm not that worried. But
Yeah.
I said last week notes. So
Yeah.
we've not received it.
Do you know something?
Let to tell me.
Do you remember that last week when we were doing? See, most of the time when I do those annotations, okay, usually I have to save them, and see, because of the light off, okay, no, I think I could still have it, okay, but so don't worry. Right after this class, then because I was busy trying to update the trial questions but please the trial question, make sure you send to everybody so that everybody's
I've
going
sent
to enjoy it.
I
Yeah.
sent it yesterday. The moment I received it, I forwarded it to everybody.
So my TA is trying to compile trial questions for multiple choice for you, and then I think with those, so that's what I've sent there is for the theory side of questions that I
Yeah,
want
that's the subjective
to keep
aspect of it.
upgrading. Yeah, so I will. He will get me the multiple choice questions, okay? Because I don't wanted to. That's why I couldn't. I paused sending the question. I wanted you to have all those trial questions at hand. Okay, so you get me the multiple choice, I think, in the course of the week, and then I'll make it available for you. But
All right, sir.
so take your
Uh,
time. No rush about that. And even here, the photosynthesis, I wouldn't even photosynthesis and respiration. I wouldn't even rush it and do it in a day. I'll break it into two because it involves cycles, and I want you to understand everything. That's more important. You getting the concepts right and making sure that because you are most of you, a number of you, I was just telling your colleagues that a number of you will go ahead and do your masters and PhD, where a number of you will be teaching at the SS level, and so you don't want to go to classroom and then disgrace yourself and disgrace me. No, I want you to go there and then deliver, even without book, without, you understand that? Like, so I want you to make sure that you get everything right and stuff like that. So please, no pressure on you. Okay, we'll finish. We'll finish everything. Okay, so don't worry.
Oh, I'm not worried. I'm not worried at all. I know what we are doing. But sir, can
Yep.
we have one other class in course of the week? I think this is going to be our
Yeah,
last meeting.
no, we'll have it. We'll have it. I mean, I don't want to take your another Sunday because I don't want to take you another Sunday because folks miss church and I don't like that. Okay, so what I would do is that we could have it we could have, because next you'll be preparing for quizzes and stuff like that. So yeah, so I'll let you have it as soon as possible. Okay, as soon as possible,
All right.
because
So we will discuss it and I'll communicate back to class.
yes, please. And then but we'll have it for last week like that, where in fact tomorrow I have a lot of quizzes. They are going to write, I think, 3 quizzes for me tomorrow. So pressure was mounting to finish some questions, and I always love multiple choice questions, so it takes a lot of time to set them. So that actually kept me so busy in the course of the week. But this week, I don't think, unless maybe you agree that we should do it tomorrow, maybe early morning tomorrow. Okay, but folks usually don't come early, and I don't want the Jesus ladies and Jesus men to miss church again, unless they decided that we wouldn't mind to take it. Because what will be left today, 2-hour meeting will be enough to finish it. Okay, and then I think fine. Okay, so it's up to you what you decide, and then if you want us to do it tomorrow so that everything will be cleared off on your shoulders, and then that would be great. Okay, but if not, in the course of the week, we must understand. I wanted the lectures that we should finish early, so that some people are slow learners. There are some people that need a little bit of time to absorb.
Yeah.
So the earlier we do it, the better. So the class will decide that the time you give it to me. Okay, if it is Sunday tomorrow, that would be great. If not, maybe you have to pick one of the evenings, and then we'll have it there. Right.
Okay. I'm not speaking for the class, but normally I would have loved if we agreed tomorrow, because the weekdays we don't know how your schedule is going to turn up like, and
Yes.
I'll be disturbing a lot of calls, just trying to get most of the calls come whilst you are in class, and I see you are worried trying to return the call. So if colleagues will agree, since we've been skipping Sundays and so we are so
Okay. All right. Argument's network has disappeared, so that's fine. Oh, okay. So we get
God has not cancelled our name from his good books. If we can take it tomorrow, as we said, is, and then the exams that follows them, because next week is practical, the following week is quiz, and then examination continues. So
yeah.
if tomorrow will be convenient for you, I believe the class will accept it, and then we take it once and for all.
Okay, yeah. I mean, I wouldn't mind to put it on tomorrow if the class agrees. I mean,
Sure.
yeah. Okay, let me give a shout out to a few people who are joining. Okay, so I can see Ben. Ben, are you there? Ben and David. I see Ben.
Yeah,
and
I'm here, Doc.
Right. Yeah, how are you doing?
I'm okay.
Oh, okay, that's great.
Yourself?
Great. Oh, by God's grace, you're managing. God has been so merciful. So we keep grinding, we keep going, keep striving. Anthony,
Doc.
how are you doing?
Doc, what about you?
This dog.
Oh yeah, we are doing well. We are doing well. We are doing well. We are not chilling like Hajiman, but we are okay.
I'm a bad boy. This dog, I'm also doing well.
Oh, we thank God.
By God's grace.
All right. Ernesto, how are you?
Dog, by grace, I'm doing well.
No, we thank God. Okay. Let me see. Messi, are you there? Messi, how are you doing? Hey, Messi, I see you. How are you doing? Nico. Hey, Nicholas.
Doc.
Yes. How is life?
I'm well.
Oh, well, we thank God.
Doc, you know what? I missed your lecture last Sunday.
Too bad. Because
I've been in darkness for the past 8 days.
Oh, sorry.
Even this morning, I had to travel over 20 kilometers to go to their life to join.
Oh, sorry.
Proper respect. Don't worry. Proper respect will come. Like
Ajuman is and he's Oyezua is up people maybe they are racist in Ghana. So
yeah, wait, there is a scam. Don't worry.
He want to do politics this month. Don't mind him. Ajimaya won.
all right. So Nico, yeah. So about you go through the notes and then in case you have any issue, please feel free to WhatsApp me and then
I will. I will.
yeah, the quizzes will be over my head this weekend. So I think I'll have a little bit of free time to respond. Okay. All right.
Okay, no problem.
All right. Belinda, how are you? Belinda, are you there? How are you doing?
I'm doing good.
Oh, well, we thank God. Welcome. We are happy to see you here. Okay, let's see who else also joined. Josephine. Okay.
Yes, Doc. Good morning.
Good morning, Joe. How are you doing?
Doc, I'm good. You?
I'm doing well, thank you. Welcome.
Okay.
Good to have you
Thank
here.
you.
Hey, Beatrice, one BB. Are you there?
Yes, please. Good morning, sir.
Good morning. How are you doing?
By Jehovah's grace, please your side.
I'm doing well by God's grace. Thank you. Erico. I
Yes, sir.
see you.
Yes, sir. Yes,
Charlie,
sir.
what's up? What they going?
We did one side. Some small washing.
Oh, Charlie, how you go do Oh, Charlie, small washing way with the take. You see, they're not gonna make you bed. So they do washing. They take the lectures. They chase you.
Yeah, double taxing.
Charlie. This be academic double track system. Charlie. How you go? Yeah, but everything cool, yeah.
Oh yeah, by grace.
Oh, you can't go. Nice. All right. So we'll wait for just a couple of people to join and we'll kickstart. We'll kickstart pretty soon. Okay. All right. Oh, one Samuelina is here. Samuelina, are you there? How are you doing? Samuelina, are you there? I see you. How are you doing? Juan Luis, are you there? Yeah, Franco is back. Yeah, Luis, what is going on? Are you okay? Are you chilling? Frank, are you happy? Franco, where is Franco?
Very, very happy this morning.
Oh, that's great. We thank God.
The grace has found me, so I have to be happy.
Oh, wow. We thank God.
I'm also happy to hear the voice I love the most.
Oh, thank you. It's good to see you here. Thank you. We can start very soon. Okay, we'll start a class very soon. Okay, as I said, even though what I have here, there are not many, but I wouldn't rush it because this side has a lot of cycles in them. Okay, let me even just go and get some detailed cycle for you. Let's say, okay, so it has a number of cycles in them. So I wouldn't rush it at all. Okay, I will take my time so that you get this concept right. Because some people would go into
Victor.
Yes.
And the market of Mona Bantem.
Hey, very much. First time I'm
I didn't go. Maybe I go West Coast. No, West Coast is here. Then I'm in Ifi. I will do West Coast camping in Ifi. We'll be there as a second. Na dokuru bi the bus ya no pana. I'm worried. I just say.
2 minutes more and then we'll start. Okay, 2 minutes more and we'll kickstart, please.
Zay.
Yep.
I think we
start?
can start. Yeah, 7:30.
Okay.
We've given enough 30 minutes. We can't be waiting forever.
Okay.
Let's start.
All
please.
right.
Thank
Great. So today is we are going to talk about one very, very important processes. Okay. That is respiration. Respiration and photosynthesis. We've put in the fermentation for a particular reason that we will know pretty soon. Okay, so respiration word respiration. Let me increase my font. Okay, the respiration is word. It's a metabolic word. Respiration and word photosynthesis. Photosynthesis are metabolic word. Metabolic processes. Okay, they are metabolic processes. It involves the building and then the breaking down of what? Of molecules. Okay, it involves the what? The building and the what? And the breaking down of what? Of molecules. Okay, and when it happens like that, I remember one of your colleagues asked question about transpiration being what he asked whether transpiration is a metabolic process. Okay, and then I said no. The reason being that transpiration doesn't involve the building and then the word and then breaking down of molecules. Okay, so here when we talk of metabolic processes, there are two things we talk about. We talk about what? We talk about catabolism. Okay, catabolism involves breaking down of molecules. And then we have what? We have anabolism. Anabolism that involves what? Building molecules. Okay, so when we talk of what? So these processes, these two main processes that I've highlighted here, they are what? They are all metabolic processes, and then they are metabolic processes because they involve what? They involve catabolism, which is the breaking down of molecules, and then anabolism, building of what? Molecules. Okay, all right, good. So we will walk through them quickly, and then yes. So let's say we have what? Okay, so let's start. Okay, so we talk of what? When we talk of energy production or energy producers. So in our biosphere, okay, almost all our energy, okay, is coming from what? It's coming from the sun. Okay, so our energy is coming from the sun. Okay, so this energy that is coming from the sun, we have the what? We have plants that will absorb the what? The sun energy for what? For photosynthesis. Okay, so and those organisms that are capable of doing that, we refer to them as what? As being autotrophic. Okay, so they are using the what? They are absorbing the sunlight. Okay, they are absorbing the sunlight for photosynthesis. So plants. Okay, plants are in this category. All right, plants are okay. So when we talk of what? Autotrophic what? Organisms. Okay, plants are in this category. Okay, then we have what? We have green algae. Okay, all right, and then we have some bacteria. What we call them cyanobacteria. Okay, and all these organisms, they have something in common. What they have in common is that they have what? They have chlorophyll. Okay, which is what? Which is a green pigment. Pigment for photosynthesis. I would say for absorbing sunlight for photosynthesis. Yep. So we have this. We have plants, green algae, cyanobacteria, and then for absorbing sunlight for photosynthesis. All right. So these organisms that they are capable of what? Absorbing sunlight for photosynthesis. Okay. These ones are what? These ones we can call them, they are photoautotrophic. Okay, because they are using the sunlight for photosynthesis. Okay, and we have plants, we have the algae, and then we have the cyanobacteria. And all these organisms, what they have in common is that they have the green pigment, which is the chlorophyll. Now we have some organisms, we can tell them they are what? They are chemo. If you look at my first name here, okay, we have other organisms that they also manufacture food, okay, but they are going to use different mechanism. Okay, so let me copy this one here and bring it here and talk about it. Okay, so they are autotrophic, all right, but in their case, they are not photosynthetic. Okay, so they are autotrophic, all right, but theirs is not photosynthetic. So when organism is capable of manufacturing the food, okay, we call you autotrophic. Okay, but if you're autotrophic, it's based on the absorption of sunlight. Okay, so which means that you are what? You are photosynthetic with autotrophs, okay? Okay, or sometimes some books will put it as the photoautotrophic, meaning that those organisms, they absorb the sunlight to make what? Energy, okay, to produce chemical energy. So these are ones that are going to absorb sunlight for photosynthesis. We have the word we call them photosynthetic autotrophs. Okay, they are capable of making their own, okay, food. All right. Then we have some, if you go to deep sea and stuff like that, in deep sea, most of the times the sun doesn't really reach there. Okay, deep sea depth, the sun doesn't reach there. And so if you are not having sun, which means that you can't perform photosynthesis. Photosynthesis is virtually impossible for you to perform, okay, when you are in an environment that there is no sunlight. So these organisms, they are capable of what? Using what? Inorganic substances, okay, inorganic substances, all right, substances like sulfur, okay, nitrogen, ammonia, okay, like that, okay, all right, nitrogen, hydrogen, okay. So they will use inorganic substances to oxygen, okay, to make to build molecules. And so when those organisms like that tend to do that, we call them chemosynthetic what? Autotrophs. Okay, we call them chemosynthetic what? Autotrophs. Okay, all right, great. All right, so some are what? Chemoautotrophs, others are photoautotrophs. Okay, so when you are using the sunlight for photosynthesis, then you are the what? You are photoautotroph. And if you are using chemical inorganic substances, sulfur, ammonia, oxygen, nitrogen, okay, to build molecules, then you are what? You are regarded as a what? Chemosynthetic or chemoautotrophic organism. All right, good. Now, then we can talk of what? We can talk of those who consume, those of us who consume, the consumers, okay? So the consumers, like all of us, we are what? We are the heterotrophs, okay? We are incapable of making our own food, okay? So if you are incapable of making your own food, then what happens is that we would depend on the food that has already been what? That has already been built, okay, by the what? By the autotrophs. Okay, so the autotrophic organisms, okay, they are going to they will make the food, okay, and then we the consumers, we are the what? We are the heterotrophs, okay. So for the heterotrophs, okay, what is going to happen is that we will have different mechanisms of acquiring the food. And then we even have mechanisms of what? Digesting the food, breaking the food down. Okay, so we have something like we have all animals we lack chlorophyll, so we can't make our own food. Okay, so all animals are what? We are heterotroph fungi. Okay, fungi like the mushrooms and co. Okay, so the fungi, they are also capable of what? They are not capable of making their own food. In fact, the fungi, we call them fungi. A specific term we give it to them is the word saprophyte. Okay, so the fungi, they are saprophyte because they take in what? They take in dead stale, okay, organisms. Okay, that's what they feed on. Okay, all right. So so these ones, the fungi, they are regarded as they are autotroph, all right. They are heterotroph, all right, but they feed on what? Dead, stale organisms. Okay, all right. Okay, great. And then we animals, animals like us, what the specific group is that for animals, we are something we use in terms of our food. In terms of the heterotroph, we are described as holozoid. Holozoid. Okay, when we say holozoid, what it means is that we take in complex food substances. Okay, we take in complex food substances that need to be broken down. All right, good. And then we have what? So we taking the complex food that need to be broken down, and then we have the chemosynthetic heterotroph. Okay, so when we talk of, let me take these pictures away. Right. Okay. Now, when we talk of the synthetic, so these ones like that, they consume. These are heterotroph, but they are consuming those food substances that have been built by the what? The chemosynthetic autotrophs. Okay, so for instance, those organisms in those fishes in deep seas. Okay, those fishes in the deep seas, of course, in the very deep ocean floor, the light is not reaching there. So usually, the type of autotrophic activities that goes on is a chemosynthetic. So the organisms that tend to feed on the chemosynthetic ones, so they are regarded as that's a chemosynthetic heterotroph. So they consume organisms that are making they are making food from the directly from carbon, nitrogen, ammonia, sulfur. Okay, so those organisms are they are chemosynthetic heterotrophs. Okay, and then we have the photosynthetic autotrophs are like us. So we consume the plants and we consume the plant, and then the plant. Okay, we will consume the plant. Okay, because so the food, okay, the rice and stuff like that we've been eating. Okay, the food that has been built by the plants, then we consume them. So those of us like that, we are we are photosynthetic heterotrophs. Okay, all right, great. All right, so so when we come to all these processes that we are talking about, okay, is basically because we need, our cells actually need energy, and energy is very, very important. Our cells need energy, okay, for us to survive. Okay, so and once energy, we require energy, which means that and if you can see from my screen here, you can see that the energy in the ecosystem here is coming from the sun. So we'll build up the glucose, the the glucose and stuff like that. And when we build the glucose, then through what? Through photosynthesis, all right? So photosynthesis will build the glucose, and then we will consume the glucose, and this glucose, another metabolic process will occur, okay? And then that is the respiration. Okay, we consume the glucose, and this glucose will be used to build up what? Build up ATP. And when we talk of the ATP, ATP is the what? The energy, okay? That's the energy currency of the cell. Okay, so when we talk of the ATP, is the energy currency. So let me put it here. So ATP. So ATP is the word energy currency, okay, of the cell. All right, energy currency of the cell. So you can see these two metabolic processes, they are a little bit antagonistic to each other. You can see that while carbon dioxide will take the heat energy, okay, the light energy from the sun, and then build up glucose, and then in the course of that, oxygen is going to be released. Then we will also take in the what? We will take the glucose in, and remember, during the photosynthetic process, what will happen is that the light will come in, and then they will use what? They will use carbon dioxide. Let me just go one step a little bit. Okay. Okay, so let's see. Hello.
Why do you refer
Hello.
to the ATP? Why do you refer to the ATP as the energy current of the cell?
Okay, energy currency. Oh, maybe mass currency. So I refer to the energy currency of the cell because our cells cannot use the glucose directly. They have to break it down into a manageable manner, and that manageable currency in the cell is the ATP, adenosine triphosphate. So when they pick up the glucose, they can't use the glucose directly. Okay, the energy currency the cell understands is the what? Is the ATP. Okay, so the glucose, when we take it in, our cells will pick up the glucose, but they have to metabolize the glucose into a state that they can actually use. And that state they can actually use is the what? Is the ATP. All right. So glucose will go there. Okay. At basic level, like at JSS level, we say, oh, energy, everything we say glucose. But at higher level, we know that yes, we'll pick in the glucose. All right. But when the glucose gets into our cell, metabolism will okay. Okay. The cellular respiration will okay to break this glucose molecule down into what? Into ATP. Okay. And in the course of that, water and carbon dioxide will be released. Okay. Please, are we clear with that? Hello.
Yes, Doc.
Please, are you clear with that? Okay, good.
Yes,
so
Doc.
let me use this blank page. Let me use blank page to actually. So if you take something like photosynthesis, okay, when we take something like photosynthesis, excuse me, let's take something. Let me put maybe 50. Okay, let's take something like photosynthesis here. So let's say this is my photosynthesis. Okay, so in photosynthesis, what are you going to have? We'll have the carbon dioxide. Okay, you have the carbon dioxide is CO2, okay, plus what? Plus water, okay, water, all right, okay, in presence of what? In presence of, and then we'll have our, so on top here, we'll have our chlorophyll, okay, chlorophyll here, chlorophyll, okay, all right, let me bring the chlorophyll down, okay, like that. So my chlorophyll is here, And then on top here, the chlorophyll is going to absorb what? Sunlight. Okay, sunlight. All right. So the requirement, we need the carbon dioxide to enter into the cell. We need water there, and then we need a sunlight. And then who is going to absorb the sunlight is the chlorophyll. Now the product you are going to get, we'll get what? We'll get glucose. We get glucose. Okay. So at the end of the day, what are you going to get? So let me put the sunlight here, the chlorophyll somewhere here. All right, great. Then what are the products? We will get we'll get glucose. Okay, glucose C6H12O6. Okay, if I should write the detailed formula here, 6 molecules of carbon dioxide, 6 molecules of water, all right, giving us the glucose plus what? Plus 6 molecules of what? Oxygen. All right. So this is what photosynthesis is giving us. Photosynthesis is giving us the food here, which is the glucose. Okay, is the glucose. All right, it's giving us the glucose here, and it's giving us the oxygen here. Okay. All right. So we have our glucose, and then we have our what? We have our glucose, and then we have our oxygen. So we have carbon dioxide and what? Water in presence of sunlight that will be absorbed by the water chlorophyll. We'll be able to convert these two molecules here into glucose and what? Oxygen. Now when you come to what? So that is photosynthesis. And when you come to cellular, okay, okay, so let's say when you come to respiration, respiration is just a reverse of what is here. So in respiration, we will use what? We will use oxygen. We will use oxygen, okay, to break down glucose, to produce what? To produce energy, okay, energy plus what? Plus CO2 plus what? Plus water. All right. So you can see that photosynthesis and what? Respiration, they are reverse. One is that it's just a reverse of one. Okay. You see, we need this one will build what? We use the heat to build a molecule. We will also use the oxygen to break down this glucose to release what? Energy. Okay, and then carbon dioxide and what? Water will be released as byproduct. Okay, so whatever that so respiration and what? And photosynthesis, they are two sides of what? Of the same coin. Okay, all right, so let's take it this way. While you are what? While photosynthesis is using the light to with carbon dioxide and water, producing the glucose and what? And releasing oxygen. We are using the oxygen to break down the glucose, release energy, and then carbon dioxide and water as byproduct. Okay, so it's just the opposite of what? Of each other. Okay, good. So let's see something here. So that's for the energy. Now, you see, when we talk of, as I said earlier on, okay, that the energy is the what? The energy currency, the currency, energy currency of the cell is the word, is the ATP, which is the word adenosine triphosphate. Okay, that is the only thing, that is the only currency that the cell understands. That is the energy that the cell understands. Now, how is this one energy? Now, what is here is what? It's a nucleotide. Okay, and if it's a nucleotide, so this is a nucleotide here. All right. And what is nucleotide? Nucleotide contains what? This nucleotide contains ribose sugar, okay, and then has what? Has nitrogenous base, but the specific type of nitrogenous base is the what? Is the adenine, okay? And then we have the sugar here, which is the ribose sugar. And then, okay, we have the what? We have three phosphate. These phosphates are what? They are high energy molecules. Okay, so they break, when you break one phosphate like that, large amounts of energy will be released. Okay, so these are three phosphate. Okay, we have the, and anytime we are building this molecule, we use what something we call diphosphate. Okay, so we have what? We have adenosine diphosphate. That's what we use. And then in the course of the process, one phosphate will join the two, the diphosphate, which is only two here. And then when one phosphate joins, then we'll get what? The triphosphate. And then so this nucleotide here is what? Is what is giving the cell its energy. Okay, so we have the what? We have the adenine here, which is a nitrogenous base. We have the what? We have the ribose here, and we have the what? We have our three phosphate. So this particular structure that we have here, that is actually the energy currency of the cell. That is the energy currency of the cell. And don't forget that adenosine triphosphate is also what? A nucleotide. Okay, it's a nucleotide. It contains ribose sugar. It contains adenine, which is a nitrogenous base, and the bond here. Okay, let me pick a marker here. The bond here, this bond here like that. Okay, this bond. There's another bond here. Okay, let me, okay. And this bond here, we call this bond here glyco. We call it glyco. Acidic glycosidic word. Glycosidic bond. It's difficult to write with my mouse. Glyco. This is better. So the bond between this ribose and the adenine, this nitrogenous base, is called glycosidic bond. Okay, and the bond between here is called what? Phospho ester bond. Please, there's no HO. Ajungo, there's no H. Okay, so phospho ester bond. Okay, so phospho ester bond is connecting here. So what we have here, this structure we have here is our what? Energy currency of the cell. It actually takes the energy into a manageable form. And that manageable form is the what? Is the ATP. Okay, good. So we have what? Now, what is the role of ATP? So as I said earlier on, okay, okay, let me project this because I think it will make, okay, good. So let's take the ATP here. Now, so we have this ATP. So this is the ATP, all right? And what happens is that when we take this ATP, okay, this ATP will break down, okay, right? An enzyme called what? ATPase, okay, ATPase, right? ATPase will break down this what? This particular ATP, and then when it breaks it down, it's going to give rise to what? ADP. ADP is what? Adenosine diphosphate. So
That is true.
instead of two, we have what? Instead of our three, it has two, and then it will break the what? It will break the inorganic phosphate off. Okay, so you can see that these reactions, they are what? As I said, you can see they are in more or less some sort of dynamic equilibrium, or they are antagonistic. Okay, let me put it that way. Okay, while this one, so the ATP breaks down to give the ADP. Okay, and then ADP also what? ADP, all right, will also add they will add the what? The inorganic phosphate to it, and I'll get our ATP back. Okay, so this is my ADP, ATP. When energy is needed by the cell, it will break the what? The this one off, and when it breaks it off, high amount of energy is produced for the what? For the cell. Okay, so you can see there, I can build my ATP by adding the what? The inorganic phosphate. Okay, inorganic what? Inorganic phosphate. Okay, inorganic phosphate, right? Phosphate to the what? To the ADP, all right, and then I'll build my ATP. Okay, and ATP, when ATP wants to release energy, when it breaks, okay, it breaks the ATP, it breaks one off, and then it will give rise to what? The ADP. All right, that's how we make our energy. Okay?
Hello, Doc.
And we can see.
Doc.
Hello, yes,
The
please.
phosphate group, are they all inorganic, or it is only the one that is broken out?
No, they are all inorganic. They are all inorganic. All phosphates are inorganic molecules.
Okay, so,
phosphate
so now that is broken out, is it going to join again before it to form the ATP so that the cycle will complete?
Yes, please. Yes, please. So you break when it breaks, it gives out energy. So like the energy from the bond. Okay. All right. So ATP releases its energy quickly by hydrolyzing this. So it breaks when the moment it breaks, okay, it gives the energy. All right, the energy will be given to the cell. Okay. So and you can see that we have an enzyme called ATPase, okay, which is catalyzing the process. So all these ones are all inorganic, what? Phosphates. Okay, all these ones are inorganic phosphate. All right. So they will join. When it joins that, then we'll build our ATP. Okay. And then when I want when I want to build ATP, I will join this one, will join this. Okay. All right. At some parts of what, when you come to the cellular response, which we'll be doing, you will see that there is what? There is also a process that will build this one, because something ATP synthase. Okay, which we shall talk about it pretty soon. So I don't want to worry your head here. Okay, so once the ATP is what? It releases its energy, it becomes what? ADP, and then we have our inorganic what? Phosphate here being released as well. Okay, all right, great. Okay, so as I've just explained, ADP is low energy molecule and need the what? Need to add the inorganic for one more phosphate to it in order to be charged as well as ATP. Okay, good. All right, so let's see something here. Now, what are the role of what? ATP in the cells, okay? What are ATP doing in our cells? Okay, good. But before that, you know what? Let me escape the screen here and then find out from you, what do you think ATP is doing in our cells? Okay, so let's see. Let me find out from the audience. Okay, let me find out from the audience what ATP is doing. Okay, so let's see. What is the use of ATP? What is the use of what? ATP in a cell? Okay, yes. Please, from the audience, please, what is the use of ATP in the cell? What is the use of ATP in the cell? Please. Please.
Doc, can we say it for building cells?
Yeah, okay, yeah, you're right. Okay, so I would say, I would because I say in the cell, so I would say that maybe building, okay, molecules in the word, molecules in the cell. Okay, ATP can be used to build what? Okay, molecules in the cells. What else again?
Sir, for energy transfer.
Okay, what does it mean by energy transfer? What does it mean?
So ATP, since it's a reaction, is a transfer of energy from one reaction to another, allowing cells to perform various functions.
Like?
Since we need ATP, how do I even put it? It's a metabolic reaction in the cell.
Okay.
And so as it does that one, it releases energy and transfer to the various side that the body needs that energy to work.
Okay. So from what you said here, because you use the word metabolism, when I bring metabolism, I have to clear off what I've written here. Because metabolism will take care of everything. So then it uses the word ATP is used for what? For metabolic word processes. Okay, processes which will involve what? Both building and breaking. So that is what building. Building is anabolism, and breaking is what?
Catabolism.
Okay. All right, good. Any other thing again? So now we can't talk of building and listening because we use metabolic process to take care of everything here. All right. Now what are other functions of ATP? Yes, other functions of ATP. Yes. Anybody from the audience? Yes, please go ahead, sir.
Hello, Doc.
Yes, please go ahead.
Doc. We say it is used in a DNA replication. ATP.
Yes, DNA replication. Okay, excellent. Okay. Now, so for instance, okay, when the DNA is going to replicate, okay, when the DNA is going to, before our cells divide, we need the DNA to make a copy of itself. Okay, we need DNA to make what? A copy of what? Itself. All right. And so for the DNA to make what? A copy of itself, all right, that is the replication. Okay, and that replication requires what? Requires ATP. Okay, so replication of DNA, well done. Replication of DNA, excellent. Okay, replication of what? Of DNA, excellent, well done. Yes, any other points? Yes, any other points?
For the cells.
Okay, all right, provide. So that's, yeah, so we know it provides energy. That's a primary thing. So we want to know, what would the energy for the cells be used for? Please, you get my question. Hello.
Yes,
Please, you get my question. Yeah, so we know that it provides energy. Okay, but what we want is that, what would the energy provided by ATP be used for? Okay, so we know for metabolic process, okay, which we are talking about what? Anabolism, which involves breaking of molecules, and we have what? Catabolism. which involving the so anabolism is building of molecules, and then catabolism, breaking of what? Molecules. And then our colleagues just said replication of what? Replication of what? DNA. All right. And this is very important process because if we need our DNA to replicate in order for our cells to divide. Okay. Okay. So, and that process is what requires what? ATP. All right. Any other process that requires ATP? There's one obvious one that we've talked about it in the past. Okay, one obvious one. Yes, yes, one obvious one. One obvious one, please. Yes, one obvious one, please. Okay. All right. So for yes.
You also said it is the energy currency.
Yes.
In the
yes, please. So yes, because it's energy currency, that is what is it is energy currency that is used for metabolic processes, for DNA replication. And what else again? Let's say you have money. So this is the money the cells staying together. Like as we work, we make money. So the cells will make the money in the form of what? In the form of what? ATP. All right. And after it makes the money in the form of ATP, we spend the money. Like when you make the money, what do? You spend the money. You use it to buy stuff for yourself, new bags, okay, new shoes, new dress, and new car, and all sort of things. So yes, we want to know what the cell is going to use the ATP for. Okay, so we talk about metabolic processes. We talk about replication of DNA. Okay, what else again? Active transport. Okay, remember active transport. Okay, active transport. Processes. Okay, all right. Those active transport processes. Remember, active transport process when you are moving what? Molecules. Moving. Okay, moving what? Moving molecules. All right, against the what? Against the what? The concentration gradient. Okay, against the concentration gradient. All right, and that is a very important process. All right, and when you are moving molecules against what? Concentration gradients. Okay, that is going to require a lot of what? ATP. Okay, so active transport process. Okay, they require what? It requires what? ATP, because you are moving molecules against concentration gradient. Okay, now what we've written here, this point we've written here, they can occur in both plants and what? Animals. Okay, metabolic processes. Okay, that's a anabolism and catabolism, DNA replication. Right, but if I should deviate a little bit, okay, a little bit, all right, okay, let's say for animals, all right, for animals, okay, remember transmission of what? Transmission of what? Of nerve, nerve, okay, impulse, nerve impulse, okay, all right, like the way I'm talking that you could hear me, that transmission of nerve impulse, okay, along the way. So impulse along the nerves, okay, requires what? Requires ATP, all right? Okay, requires ATP. And then we can talk of what? We can talk of muscle contraction.
Contraction.
Muscle contraction also requires what? ATP. Okay, so the first 3 points that I've made here, these are all what? These are all metabolic processes that will occur in both plants and what? Animals. Okay, but for animals alone, we can talk of what? We can talk of the transmission of what? Of nerve impulses, and we can talk of what? Muscle contraction. But these processes all require what? They all require energy, right? In the form of what? ATP. Right. Good. So,
hello, sir.
Hello, Ajuman.
So, sir, what will you put enzyme production? Is it for both?
Enzyme production will be under metabolic process.
Okay.
Enzyme production is enzyme even the metabolic process themselves, they even require enzyme. So for enzyme is how would I put it? When you talk of enzyme, metabolism and enzyme, like enzyme is the pillar, like the central pivotal, okay, is the step, okay, it's the pivot of metabolic process, okay, all right. For when it comes to enzymes, okay, so enzymes are the what? They are the pivots, okay? They are the pivots of what? Pivots of metabolic processes, okay? They are actually they are in the central position, okay? All right. Enzymes are in the central position, all right? Great. Okay. So any headache so far? Any headache so far? All right. So now that we know what the ATP is doing in cells, okay? Now that we know what the ATP is doing in cells, okay? What we're going to do is that I want to see some pictorial overview. Now, if you look at my picture here, okay? Look at my picture here, all right? You can see that we have the what? Let's take it this way, that we have the mitochondria here. So this is the mitochondria. Okay, so whether plant or animal cell, the mitochondria is the what? Is the organelle within which large amounts of what? ATP is going to be what? Produced. Okay, so the ATP is produced there by through the cellular processes. Okay, and then when the ATP is generated, right? Then we have the what? When the ATP is broken down, okay, then it will release the what? Release the phosphate, and during the release of the phosphate, we have the what? We have the inorganic phosphate will be released, and then the what? We have the ADP, which is the adenosine diphosphate. Okay, all right, adenosine diphosphate. All right, okay, great. But this one, I don't want to worry your head with this image because I'll be going into much more detail along the line. Okay, all right, good. So now let's see something. So we're going to talk about what? We're going to talk about the cellular respiration. Okay, so cellular respiration, okay, clearly as we can see here, it's a metabolic what? Process. Okay, it's a metabolic process. Okay, it's a metabolic process. So it involves the word that it's and specifically catabolic. Okay, it's not only just catabolic. Okay, some molecules will also, but because we use the catabolic because you can see that glucose, the main reactant in this reaction is the what? Is glucose. Okay, is the glucose. All right, glucose is the main reactant in this reaction. Okay, and main reactant in this reaction is going to be what? Is going to be broken down. Okay, all right, it's going to be broken down to release what? Energy. Okay, so glucose is the main reactant in this reaction, and that is going to be broken down. So that is why we are referring to it as what? As a catabolic what? Process. Okay, because glucose is the main. Glucose is the what? Is the main. Okay, starting what? Starting reactant. Okay, all right, great. So here in this process, what is going to happen? It's to involve the what? The breaking down of what? Of glucose. So the energy hiding in glucose, okay, and then to make what? To make ATP, which is the energy currency. I call it the word, the energy. Okay, energy what? Energy currency of the cell. All right, good. And so we have what? So energy currency of the cell, all right? And then, so when you come to this sort of what? This sort of cellular respiration, we have two main parts, okay? We have two main parts in the cellular respiration. We have the part that requires oxygen. If we require oxygen, you are the what? You are referred to as the what? The aerobic, okay? So as part of the cellular respiration, okay, we have what? Two main parts of it. We have the part that requires oxygen, that is the aerobic respiration, and we have the parts that do not work. We have the parts that do not require what? Oxygen. Okay, so this is that. So we have the part that requires oxygen, and then the part that do not what? Require oxygen. Okay, like that. So we have the aerobic respiration, okay, and then we have the what? We have anaerobic what? We have anaerobic respiration. So the anaerobic part doesn't require oxygen, and then we have the aerobic part requiring oxygen. So anytime we talk of what? We talk of the cellular respiration, cellular what? Respiration, okay, we have two processes that actually come together. We have the what? We have the aerobic, okay, the cellular respiration has two components, aerobic and what? Anaerobic. Anaerobic, okay, all right, we have the aerobic and anaerobic, all right, okay, coming together, okay, and then that will release energy, okay, all right. So quick one, the anaerobic, the part that doesn't require what? The part that doesn't require, okay, the part that doesn't require what? Oxygen, okay, occurs in the what? In the cytoplasm, okay, occurs in the cytoplasm, all right, and then the aerobic part occurs in the what? In the mitochondria, okay, so the mitochondria here, all right, that's where the aerobic part of the respiration tend to occur, okay, all right, great. But before that, there are some terms we need to understand, okay? Here, I don't want you to see the slide here. Let me explain it here for you, and then after that, I will jump the slide for you to see it. Okay. Now, let me increase. Okay. Now, when we have we have some chemical reaction. Hello, yes, please.
Please repeat. You said anaerobic respiration occurs at where?
Anaerobic occurs in the
cytoplasm.
Please take your time. I want to hear the thing.
So anaerobic occurs in the cytoplasm, while the aerobic occurs in the mitochondria. Okay. All right. So anaerobic occurs in the cytoplasm. It doesn't involve oxygen. Okay, that's anaerobic. It doesn't involve oxygen. Occurs in the cytoplasm, and then very small amount of energy. Okay, or you can just very small what amounts of energy. Okay, small amounts of energy is produced. Okay, so let me do it here. All right, so anaerobic respiration, anaerobic parts of the respiration, okay, occurs in the cytoplasm, okay, and then small, relatively very small amounts of what? Energy is produced, and then it doesn't require oxygen. All right. Okay, so that is it here. And then when we come to the aerobic part, the aerobic, okay, the aerobic part of the cellular respiration. This one requires
Oxygen.
Large amounts of
energy.
Large amounts of
And then
energy is produced.
Hello, Doc.
Hello, please.
Please, the second point, the small didn't come.
Oh, okay. Oh, sorry. Thank you. Thank you. Great. And then large amounts of what? Large amounts of energy is produced. So the amount of energy here I can put into bracket ATP.
Happens in the mitochondria.
In this one, large amounts of energy is produced. Let me make it this way. This way. Okay, it's produced. And then the last point here is that okay, it's okay in the mitochondria. Okay. Singular is June. Okay. All right. So these are two parts of what? Cellular respiration, the anaerobic, and then we have the aerobic. The anaerobic occurs in the cytoplasm. Small amounts of energy is produced. It doesn't require oxygen. This one requires oxygen. Large amounts of ATP is produced. It occurs in the cytoplasm. It occurs in the mitochondria. Please, are you clear? Hello, please.
Please, when you read other books, they measure the energy. They
said
what they call the cellular is aerobic is 38, and then that of the anaerobic is 2. So I wanted to know mechanism for which they use to measure the amount of energy.
Oh, we shall come to it. We shall come to it. We are building. This is overview. What he says is overview. The anaerobic and all this, so we'll go into a little bit detail. We shall come to it. Okay. So we are building small. Okay. Please, are you with me?
Yes, please.
Okay. All right. Thank you. Okay. So the next one I said I want you to see first. I want you to get this one. There's some reaction recall. I know you are doing chemistry, so definitely you might have heard it before. So there's a reaction we call redox reaction. And when we talk of redox reaction, okay, it's a reaction that has two parts. Okay, the reaction has two parts. In fact, this word redox itself is actually amalgamation of word of two terms. Okay, that is a reduction and a word and oxidation. All right, I'm sure you might have heard this thing in your chemistry class before. Okay, good. Now this, so they took this one, this and that, to give it the name of the reaction. Okay, they took the red here, the red, and then they took this one, the OX, and then they gave the name. Okay, so this reaction, the two reactions will occur at the same time. So the redox reaction contains reduction reaction as well as oxidation reaction at the same time. Okay, now so let's see what does it mean. Okay, now when you talk of each of them can be defined in 3 ways. Okay, each of them can be defined in 3 ways. So I will give all the definition to you, or I'll do one and you will automatically do the other one. Now, these two reactions, they are antagonistic. The antagonistic means that when this one is down, this one is up. When this one is gaining, this one is losing. Okay, so that's what the antagonistic. Okay, all right. So they are in two sides of what? Of the same coin. Okay, and the two of them must be present for the what? For the redox reaction to occur. So when time we talk of redox reaction, it is basically reduction oxidation reaction. That's what it means. So redox reaction is simply reduction oxidation reaction. Okay, reduction oxidation reaction. Okay, so let's see something here. Let's see something. So let's start with reduction. Okay, let's start with reduction. Okay, now when we talk of reduction, reduction is what? Reduction is can be defined. I'm going to define reduction in three ways. I will do the reduction and you will define oxidation for me. One of the ways we can define reduction is what? In terms of what? Electron. So in terms of electron, in terms of electron, we can define reduction as what? As gaining of electrons. Okay, gaining of what? Electrons. So meaning that when a molecule is there and the molecule gains electrons, we will say it has been what? Reduced. So let's take it this way, that I have something like copper here, copper 2+ like this, and then I add 2 electrons to it like that. Okay, and I end up getting copper. Okay, so this is copper ion. All right, I've added 2 electrons to it, and then I've gotten the copper element. Okay, this is copper ion. So what I've done here is reduction. Okay, what I've done here is what? It's reduction. The molecule has been what? Has been improved. Okay, so one way you can define reduction is the word gaining of what? Electrons. Gaining of electrons. Okay, good. Another way we can define reduction is in terms of what? Hydrogen. So in terms of hydrogen, we can also say that when it's addition of what? Addition of hydrogen. All right. So when you add hydrogen molecules to something, we say that that substance has been reduced. Okay. So let's say I add hydrogen to, let's see if I have something like this. I have, let's say, sulfur here, and I add 2 molecules of hydrogen to it, and then I end up getting hydrogen 2 sulfide. Okay, like that. We can say that the sulfur has been what? Reduced. The sulfur has been what? Reduced. Okay. All right. The sulfur has been reduced. All right. Why? Because they've added sulfur to the what? They've added hydrogen to the sulfur. So when you add hydrogen to the sulfur like that, we can say that this is also a form of what? A form of reduction. Okay, so reduction can be defined in terms of electron. In terms of electron, that's the gaining of electrons is regarded as what? Reduction. Or addition of what? Hydrogen to what? To sulfur. Okay, or addition of what? Hydrogen to a molecule is also described as what? Reduction. So here you can say that the sulfur has been what? Has been reduced. Okay, good. And then the last one here is that you can define it in terms of what? Oxygen. In terms of oxygen, we will say that addition of what? Removal of oxygen. Removal of oxygen from a molecule okay, is regarded as what? Another example. For instance, if I have something like this, for instance, if I have something like this, let's say I have Cu, let's say copper oxide. Okay, copper oxide, which has oxygen here, and then I remove the copper oxide like this, and then it becomes Cu. Okay, you can see that I've removed the oxygen from copper oxide. So removal of oxygen is also regarded as well as reduction process. All right. Now that I've done this one for you, the opposite of everything I've talked about here, okay? The opposite of everything I've talked about here is what? Is oxidation. So now your task is simple. You are going to write 3 definitions for me. I'm going to write 3 definitions for me. Okay. So let me give you 1 minute. Okay, let me give you 1 minute. All right. Okay. Oh, okay, 2 minutes. You're going to write definition of what? Oxidation in terms of what? In terms of what? Electron. And then write another one in terms of hydrogen. Write another one in terms of what? Oxygen. Okay. So please, your time starts now. Okay. Everything you've written here, the opposite of it is for oxidation. So please write the definition for me to see. And if possible, write the word, a reaction. So for instance, if you want to write the reaction here, all right, you can see that here, I can reverse this process here. I can do this, so I can bring this one here like this. Like that. Okay. I can bring this one here like that. Okay, like that. You can see that electron is being what? Removed. You've removed electron from the copper. And when you remove electron from the copper, that's what happens. Okay, so this is what is here is an example of what? Oxidation. So oxidation is what? Removal of what? Removal of electrons. Okay, or loss of what? Loss of electrons. All right. So please write the rest for me. You have 2 minutes. You have 2 minutes. Write it quickly for me. Yes, 2 minutes. 2 minutes. Yes, you have 2 minutes. Okay, everybody write it in terms of hydrogen and in terms of oxygen. Okay. Stephanie? Please, Stephanie? Hello, Stephanie, are you done? Okay, so let's see. Folks have sent some of their writings here. Okay.
Hello, sir.
Hello. And yes, Madam Julie, let me hear from you.
So we have addition of hydrogen, addition of oxygen to a molecule.
Excellent. Okay, good.
Then removal of hydrogen.
Excellent. Okay. I'm
And
sure
then
that's what.
the last one was loss of electron.
Excellent. Okay. Beautiful. Well done. Okay. So so basically, I hope you all agree. I hope you all agree to what Julie just said.
Yes, Dog.
Okay, thank you. All right, good. So that is great. So the next thing, so this is basically what is there. Okay, this is basically what is there. Okay, so I don't want us to go into this thing in it anymore. Okay, so let's see. So now the last part here is the most what is important to me. Now, so in cellular respiration, okay, the cellular respiration is actually a redox reaction. And one beautiful thing about redox reaction, if I should go back to this, so if I should go back to what I have here, you see, if this side, so if this side is if there is reduction, okay, oxidation on one side, the other side is undergoing what? Reduction. Okay, so for instance here, you can see that here on this side, if I add the electron to this, I'll get copper. So here on this side here, we can say that this is what the copper 2 oxide is losing what? Electron. So that is what that is oxidation. Now the reverse of it is what? Reduction. Okay, if like the reverse of it here is reduction. Okay, all right. So don't forget that. Now, so cellular respiration is what? It's a redox reaction. Okay, what does it mean? So it involves the what? It involves oxidation and reduction of what? Of molecules. Okay, it involves what? Oxidation and reduction of what? Of molecules. Okay, it involves oxidation and reduction of what? Of molecules. Okay, it involves oxidation and reduction of molecules, and the two processes are occurring at the same time. So the reduction and what and oxidation reactions, they are occurring at the same time simultaneously. Okay, so in cellular respiration, reduction reaction, okay, reduction reaction and oxidation reaction are occurring at the same time to synthesize the what? ATP, okay, for the cell. Okay. Are we clear with that? Okay, good. All right. So basically, this is the picture. The picture you are seeing here. No, let me put it this way. The picture you are seeing here is a cellular. It's a cellular respiration. What you are seeing here, this is cellular. So the picture you are seeing here is a cellular what? Respiration. Okay. It's a summary of what? Cellular respiration. All right. So cellular respiration has three main processes or three main stages. What are they? We have glycolysis, okay? Glycolysis, okay? And then we have what? We have Krebs cycle, and then we have something we call electron transport chain. Okay, so cellular respiration has three main processes. What are they? Okay, so let me go one more step here. So we are saying that cellular respiration, okay, has three main processes. What are they? Okay, what are they? Okay, the cellular respiration has three main. The cellular respiration has three main processes. What are they? We have the glycolysis. We have the glycolysis. Okay, followed by what? Followed by Krebs cycle, followed by electron transport chain. So these are the three main processes. Okay, three main processes that are involved in what? In cellular respiration. All right. Now these two here, okay. So now that you know this, so let me break this thing down. Now this one here, this part, now this part here, the glycolysis is the anaerobic part. Okay.
Hello,
Wow. Hello, please.
Please, the slide you showed first before this one.
Yes, please.
Per the diagram, when we move from glycolysis, we move to transition reaction.
Yes, I will talk about it. So that's it is transition. It's no main.
Oh, okay.
Like intermediate. Do you understand that? It's intermediate process. So we'll talk about it. We'll talk about it. But the main one, most books that you see, they will not even mention this one.
All right.
We'll talk about it. But the main one is this, that.
Okay.
So this one is just a transition. When you see something is transition, like something like transient process, it's just let me yeah, it's like basically I want to give simple analogy that let's take the transient process. Okay, let's say like courtship. Okay, courtship behavior is more of a transient. Is that not? It's a transient to marriage. Is that not it? Because it doesn't take that long. Okay, it's only Ajiman who will court somebody's daughter for 10 years. Okay, but courtship behavior, courtship
Today started day 2.
Courtship is like 1 year, 2 years.
Lord,
It's transient.
Lord that asked the person courted for almost 5 years. Lord. So he was not he was even more than the transitional thing. Lord.
All right. So it is expected. So it's just a brief, okay, it's a transition. Okay. But the main ones are the word glycolysis, Krebs cycle, electron transport. So these are the three main processes. Okay, all right. So I want to throw more before I'll show the diagram again. So we have the word. So the glycolysis is the anaerobic part. So this is the part that doesn't require no oxygen. Oxygen is required. Okay, so that's the anaerobic part. And then the words, so the Krebs cycle. So this one, remember I told you that the anaerobic part occurs where? Occurs in the cytoplasm. Cytoplasm. So we know that the glycolysis is going to occur in the cytoplasm, where the Krebs cycle plus the what? Krebs cycle plus the electron transport chain, okay, will occur in the what? In the that's the aerobic. Okay, so that is the aerobic part of the cellular respiration. That's the aerobic part of the cellular respiration. And that one occurs where? Occurs, okay, so the two of them, the two occur in where? Okay, in the mitochondria. Okay, all right, and that is the word. That is the okay. So so here glycolysis. Okay, the glycolysis is here, and then the Krebs cycle here. They are so the glycolysis is the word. Is the doesn't require oxygen, and here oxygen is what required. All right, good. Okay, so this is quite a little bit overview of it. So the glycolysis is when we talk of the anaerobic, is the glycolysis we are actually referring to. Okay, and then the aerobic we are referring to the Krebs cycle, and the what? The electron transport chain. They actually make the aerobic. So together, that all these three steps, okay, together make up the what? The cellular respiration. Cellular respiration. Okay, please, are we clear with that? Are we clear with that?
Yes,
okay.
No.
Good. So now when I go back to this, my diagram, when I go back to my diagram here, okay. Now, so in this particular diagram, you can see the Krebs side, the glycolysis being pointed at the word at the cytoplasm here, and then we have the transition reaction occurring in the word in the mitochondria, in the mitochondrion, and then the you can see the Krebs cycle also in the mitochondria, but you can see they are pointing to very different positions, and the electron transport occurring also in the mitochondria. Okay, so these are very simple processes that we'll be taking them one by one and talk about them. Okay, so the most important thing is that you appreciate this sequence. Okay, and then before I begin to talk about it. Okay, great. I don't want to go into detail of the transition here because I'll talk about them in the latter parts. Okay, but we know that there is going to be a transition reaction. Okay, so glycolysis, Krebs cycle, and the electron transport chain, and we have the transition reaction here. Okay, which will usher it before you get into the Krebs cycle and then the electron transport chain. Okay, all right. Okay, good. So that is that for this one. Now let's talk simply about what? Let's talk simply about we are going to talk about the what? Now when we talk of the glycolysis, what is it? When you talk of the glycolysis, what is it? Okay, when you say from the name, what is it? What is it? I'm sure, definitely, I know you might have heard it before. What is glycolysis? When you talk of glycolysis, please, what is it? Please, when we talk of glycolysis, what is it? Yes, can somebody help me? When you say glycolysis, what is it? Please.
The breakdown of glucose.
The breakdown of glucose. Excellent. Okay. So glycolysis is the breakdown of what? Of glucose. Okay. And when we say breakdown of glucose, what does it mean? Okay, what does it mean? Yes. What does it mean?
That's glucose metabolism.
Glucose metabolism. Excellent. Any other point? Any other point? Any other point? Yes. Yes. Any other point? Any other point? Yes. Any other points there? Yes.
Okay, sir. Can you also say that it releases less amount of energy since it falls under anaerobic respiration?
since it falls under anaerobic respiration, okay, that is good. That's also a nice point there. Any other point? Any other point? Yes. Okay. Yep. Any other point? Yes. Okay, please, just a minute. Okay, somebody is knocking at the office door. So just a minute. Let me just and then come. Just a minute. Okay, please. Okay. All right. So yes. Yes. Okay. Go ahead. Okay. Okay. So, Ajima, you made a point. Please come again with your point. Ajima, come again with your point.
What I said was it releases less amount of energy since it falls under anaerobic respiration.
It releases less amounts of energy. Okay. All right. Not bad. Okay. What else again? What else again? What else again? Yes. What else again? Yes. Now you can see there's this term here called that's hexagonic. What does it mean? When you say hexagonic reaction, what does it mean? What does it mean? Yes. When you say reaction is hexagonic, what does it mean? I don't understand that.
Sir,
Please. Yes. What
sir,
does it mean? Yeah. Yes, please. Go ahead.
to break down fats and proteins into smaller particles.
Okay. Now, so breaking down glucose. Hello. Yes, please. Let me hear it from you again. Yes. Any other point? Yes. Please go
It means
ahead.
energy is released. The release of energy.
It means energy is released. Any other point? Yes. Any other point? When you say it's what? It's a hexagonic. What does it mean? Hexagonic. I don't understand. It means release of energy. Who agrees with our brother? Yes. When you say something is hexagonic, what does it mean? Okay, so now that we have a term like hexagonic here, let's see something here. Let me see. Let's see. Let me take this. Okay, let me open. Okay, so let me enter into another slide. Okay, let me put this one here. All right, let's see. Now our brother said it's what? It means releasing of energy. Okay. Yes. Please, how many people agree with him? How many people agree with him? Yes, please. Yes. Yes. So what should be the opposite of this one? What should be the opposite of this one? What should be the opposite of this one? So, which means that anytime we say a reaction is what? It's hexagonic, it means that the reaction, hexagonic, what it means here is that the reaction releases what? Releases energy. All right? Are you there? The reaction releases what? Energy. Okay? And when it is what? So what will be the opposite of this one? The opposite is going to be what? Yes. And somebody
Gaining
help
of energy. Gaining of energy.
Yes. And then the what? And that good. So we can get
Absorption of energy.
good. That's and that is going to be what? That's endogonic. So endogonic means that when we say reaction is endogonic, it means that it's what? It requires what? Energy input. Are you there? Please, do you get it? Are you with me?
Yes, Doc. Yes.
Yeah. So when you input energy, we say that the reaction is what? Hexagonic. Endogonic. And then when you release energy, we say the reaction is what? Yeah, when you release energy, you say that because you are giving the energy to outside, okay? All right, so exa talks about outside, and enda talks about what? Inside. So here you are giving what? Giving out what? Energy, all right? And here you are giving out what? Energy, okay? And here you are taking in energy. Please, do you get a difference? Okay, please, are you with me? Do you get a difference? Okay, great. So what it means here is that when we say a reaction is what? Hexagonic, it means that the reaction will give out energy. And when we say it is endagonic, it means that the reaction will require what? You rather add energy to the reaction. The reaction rather takes in what? Energy. All right. Are we clear with that? Okay. All right, good. So this particular reaction, you see, it's a word. It's what? It's hexagonic. Okay, hexagonic means that at the end of the reaction, energy is going to be what? Produced. Okay. All right. The energy is going to be produced. So at the end of the reaction, energy, okay, is going to be produced. Okay, good. And then I've talked about glycolysis already. That okay, so it occurs in the word cytoplasm. Okay, and you can see that cellular respiration from this my image, you can see cellular respiration has what? It starts with glycolysis and ends with what? Electron transport chain. The glycolysis part doesn't require what? It doesn't require oxygen. So that part is what? That part is aerobic process. Sorry, it's anaerobic process. And then from the transition all the way to electron transport, that one what? It requires what? That part, okay, requires the presence of oxygen before the process what will be triggered. Okay, so that part is the what? Is aerobic process. Okay, so cellular respiration, as I said earlier on, has two components. Okay, has the what? Has the aerobic part and has the anaerobic part. Okay, the anaerobic part is the glycolysis, and the aerobic part we have the transition. Okay, well, the main are the Krebs cycle and the what? Electron transport chain. Okay, all right. So what we are going to do is that we're going to talk about our this side. After all this side here, we are going to elaborate them in detail. I think we've shown with diagram. So let me just give overview of the glycolysis, and then we can give ourselves a break. Okay, so that couple of folks who came early could take some sort of tea and then stretch a little bit, and then we'll come back. Okay, all right. So let me talk about the glycolysis. Okay, first, and then I've given the overview already, but let me talk about it here. So let me just get to my one slide here, like that. Okay, and then let me use it to explain. So glycolysis. Okay, good. Now, let's say, all right, good. So let me put it this way. All right, here, like that. Okay, so let me draw a cell. Okay, so let me go to take a marker here and draw. So now, let me draw my cell here. Let me draw, okay. Oh, okay, let me pick green here. It's plant, so let me draw my beautiful plant. Oh, look at what I've drawn. So let me draw my beautiful plant cell here like that. Okay. Okay. Now, after drawing this, my picture, so let me put a very big vacuum here. All right. So I have my mitochondria here like this. Okay, like a sausage-like stuff in there. Sausage-like stuff in there. All right, then I have my what? This side is the cytoplasm. Okay, inside the cytoplasm here. Good. So what is going to happen? Now, so glycolysis starts. Okay, let me go and come back here, and then this place will be open. So now let me put let me increase. Let me take 40 for now. So glycolysis, the word glycolysis simply means what? The splitting of glucose. Lysis, the word lysis means breaking. This one. The word lysis, this side. Okay. Splitting of what? Splitting of glucose. Okay. Splitting of glucose into what? Okay. Into two pyruvate. Okay. All right. With the release of what? 2-pyruvate. Now remember, glucose is what? If the formula I wrote earlier on, okay, if you remember, glucose is what? C6H12O6. This is the formula for glucose. So glucose contains how many carbons? It contains what? 6 carbons, right? The carbon there. So glucose contains 6 carbons. Six carbons, right? Okay, like that. So let me keep all these ones in bracket. So it's the breaking of the what? The splitting of the glucose into what? Into 2-pyruvate. Okay, pyruvate, or you can also say pyruvic acid. 2-pyruvic what? Acid. All right. Now the pyruvate, okay, okay. And then when it happens like that, there will be release of what? There will be release of what? Of ATP. Okay. There will be release of 4 ATP. Let me be specific. Okay. 4 ATP. And then another substance that will be released as what? NADP will be released as well. Okay. All right. And then so we have what? So here you can see clearly that in glycolysis, all right, we are having what? We are having 2 pyruvate molecules. Okay, so let me come down here. Okay, 2 pyruvate molecules giving us what? Giving us so let me put glucose. Okay, so glucose here, glucose like that is there. Will break down, and remember, glucose is a 6 carbon molecule. Will break down and give us what? Give us 2-pyruvate, and then give us what? 4. Okay, 4 what? Okay, 4 ATP plus what? Plus what? Plus 2 what? 2 NADP. All right. Okay, so this is the reaction. Okay, this is actually the reaction of glycolysis. Now, let me, you see, I wrote 4 here. There are reasons that I wrote all these ones here. Now, on top, before this reaction will go on, this reaction, let me go next step here. Let me do like that. Okay, so let's say like that. Okay, let me come here like that. This reaction requires 2 ATP. Before this reaction will occur, okay, 2 ATP, you have to add 2 ATP, okay, for the reaction to occur. So I want to see, let me see, let me reduce the font of text. This one. So here, before we can break the glucose down, what do we need? We need 2 ATP to come. Okay, we need 2 ATP, and at the end we will get what? We get 4 ATP, like that. 2 pyruvate and 2 NADP. All right. So let me elaborate things okay, a little bit. So glycolysis is the splitting of glucose, okay, to 2 pyruvate, okay, 2 pyruvate, 2 molecules of what? Pyruvate, 4 ATP, and 2 molecules of what? NADP. All right. Oh, look at me. I say NADP. 2 molecules of what? NADH. The NADP is in photosynthesis. Sorry, this one. NADH. Okay. 2 molecules of what? NADH. Now, let me throw more light on it. For the last time, glucose glycolysis is a splitting of glucose, okay, into 2 molecules of pyruvate, 4 molecules of what? ATP, and 2 molecules of what? NADH. Now,
ADP means?
Good. Nicotinamide adenosine. Okay. All right. Are you there? Nicotinamide adenosine dinucleotiphosphate. But don't worry, we shall come to it. We'll elaborate it. Okay. We'll elaborate it very soon. I'm going to talk about each of them. Okay. Are you with me? Hello. Hello.
Hello.
Yeah, I'm going to talk about each of them here. I'm going to take them one by one and talk about okay each of them. I just want to give you the overview here in this reaction. Okay, then we are going to just take each of them right now and then before we go on break. Okay, I just want to break this one down before we go on break. Okay, please just give me 5 minutes and then okay. So now let's start. Now we are start on this reaction. Now glucose is going to break down to get what we have here. Okay, and so the first point I want you to note down is that before the breaking down of the glucose, so the glycolysis, we said that glycolysis is what? It's hexagonic process. It requires what? It gives energy. But have you also realized that glycolysis requires what? Energy. Please, have you seen it? Please, can you see that? Before glycolysis will occur, so glycolysis is both what? Gonic as well as what? Endagonic. Please, do you understand?
Yes.
Good. So that's why I said I'm going step by step. So from here, we can see glycolysis. Glycolysis is what? From what I've just I have on my board, you can see glycolysis is both hexagonic, okay, and what? And endagonic, okay, process. Because It's hexagonic because it is releasing what? So let's see. Let's take one. Okay, hexagonic. So it is hexagonic because it is releasing energy. Are you with me? Are you with me?
Yes.
Good.
Yes, please.
All right, good.
Yes, please. We are with you.
And then it is what? Then the next point, it is endagonic because it requires what? It requires what? It requires ATP. Okay, all right. So glycolysis is both what? Hexagonic and what? Endagonic. It is hexagonic because at the end of glycolysis, energy will be what? Energy will be released. Okay, in the form of what? ATP. Are you with me?
Yes.
Please,
Yes.
are you with me? Good. All right.
Yes.
Good. Excellent. Good. Now, the next thing is what? The next thing I want you to, so here, right now, the key take-home message now is that glycolysis is energy dependent what? Energy dependent process. Okay, why? Because it requires what? It requires because it requires what? ATP. Are you with me? Hello?
Yes.
Hello, Doc.
Good.
Hello, Doc.
Hi.
Hello, are you with me? Are you getting
Please, we
the
are
point
with you.
here? Are you getting the point?
Yes.
So
Yes, please.
glycolysis, even though the process will give energy, but before the process starts, it requires energy. The process itself requires energy. So it is energy dependent process. Why? Because we require 2 ATPs for the reaction to start, for the reaction to occur. But when it occurs, it will give us what? 4 ATPs. So glycolysis is energy dependent, is ATP dependent, but oxygen independent. Process. But oxygen what? Oxygen independent process. Okay, all right. It doesn't depend on oxygen, but it depends on what? ATP. There's a question I'm expecting someone to ask me right now. There's a question. Glycolysis require ATP, 2 ATP for the reaction to occur. And when it finishes, it gives us 4 ATPs. All right. And we know glycolysis is what? It's anaerobic process. Okay, it's anaerobic what? It's anaerobic process. Okay, process, and therefore does not what? Does not require oxygen. Does not require what? Oxygen. Okay, so glycolysis doesn't require oxygen, doesn't depend, okay, upon oxygen, doesn't depend on oxygen, but it depends on what? ATP. So somebody should ask me. There's a question I'm waiting for somebody to ask. Yes, which question? Now the question is,
So the question is why? Why does it not require oxygen?
Oh, okay. Well, the process, oxygen is not really needed in the process, when we go through the steps, there's no oxygen needed. But the question I was expecting is this: that you are going
So,
to
sir,
go ahead. I'm listening.
so the 2 ATP
Mm-hmm.
that started the reaction to
Mm-hmm.
get the 4 ATP
Mm-hmm.
after that is as a product, or after this thing, during after the reaction, how did the additional 2 ATP come about?
All right, good. That one we'll get it very soon. But the question I'm asking is that why the ATP at all in the reaction? You yourself, you are coming to generate ATP. So why do we at all give you, just like somebody telling you is that oh, I'm coming to give you food. I'm coming to give you food. And then he says that before I give you food, give me food first. Is it Ajima, where in the Bible? I think was it Elijah or Elisha? Which one that the prophet got there asked the widow to give him food first before? Which
This one,
one is that?
I don't know. I don't know this one.
Oh, it's
Elisha.
Elisha
okay.
and
Elijah. Elisha and
Victor,
Elijah.
remember, I am a priest, eh? I'm an ordained minister. If you don't know.
Okay.
Yeah, Elisha
That's why.
or
That's
Elijah.
why. Is it Elijah or Elisha?
Elisha.
Elisha and the widow. Elisha and the widow.
Okay. Oh, okay. Good. So he got there and he says that give me the food first. Okay. Good. So this is what glycolysis is doing. That before I do it, before I produce my ATP, give me the ATP first. Now, so which means that the last statement I want to make here before I go to that is I'm still on this side though. I'm still on this side. Now, so which means that at the end of glycolysis, okay, produces a net ATP of what? Of what? 2. So it's like what? The government. You know, the government pays me. Okay? He pays me. But when he pays me, I do what? I pay tax.
Tax.
You understand that? Okay? So the tax the government is taking here is the what? Is the ATP here. All right? So at the end of the day, when they deduct all my income tax, then before I'll get what? My net salary. Okay, so which means that from here, you can see clearly that at the end of glycolysis, the net energy production is not 4, but it's rather what? 2. Because you are spending what? You are spending 2 to get 4. Okay, so at the end of the glycolysis, I'm getting what? I'm getting a net ATP of what? A net ATP of 2. Please, do you get it? Please, do you get it?
Yes, Doc, we get it, but my confusion is it is the same glucose that is going to be broken down into the ATP.
Exactly.
So why the ATP again must be added to the glucose? Which is
Beautiful. That's the $1 million question I'm asking. I'm waiting for somebody to ask. So finally, you have asked it. Okay. So now that you've asked it, I have no choice than to explain. But before I explain, we are still on this side, though. We've not crossed to this side. So here, we are here. Glucose. You yourself, you are saying the government. You yourself, you pay, you are the one who is paying my money. And the one you pay me, you say that before I pay the money, I have to pay something small. Okay, it's like somebody going to look for a job and they're asking, hey, connection man will tell you that bring this amount of money if I give you the job. Like that. Okay. So now before I go to that one, we know that glycolysis will produce 2 pyruvate, 2 ATP, and 2 NADP. Okay, good. That is, we know that one, that part of the reaction, we know that one. So we've not crossed the boundaries here yet. We know there's the first step. Everything will be explained to you. Okay, no problem at all. Now, then the next one. Okay, the next one is that we have the word. We have the glycolysis is both what? It's both hexagonic and endagonic. Why? Hexagonic because it releases energy. At the end of the reaction, energy will be produced. And we can see here, energy is produced. It is endagonic because it requires what? Energy input as well. And that is here. And that is the $1 million question. Why that ATP in the first place? Okay, which we will answer it very soon. So we know that glycolysis is energy dependent process. Okay, because it requires what? 2 ATP. But we know glycolysis is oxygen independent because it's what? It is anaerobic process. Okay, it doesn't require oxygen. Okay, all right. Okay, so let's see. Now that $1 billion question has been asked, now let me get a new slide and let's see. Let me use it to explain this. Hey, 2 minutes. Oh, I've kept you people beyond the time. Okay, so 2 minutes. Now, quick one. Let me go and take this my image here and bring it here. Okay, now my image is here. All right. Okay, there is this adage that people say, marriage door, they don't close it. So if you want to leave, you can go out. That's one of the funny adages, that people say, that marriage door, they don't close it. So if you want to go out, you can go out. Why am I saying this? I have my glucose here. Now, the glucose must be absorbed into the cell. So the glucose will go into the cell, right? Okay. So the glucose will get into the cell. Must be what? So will be absorbed, okay, into the what? Into the cell. All right. Now, so let me go and pick a, and this glucose is what? C6
H12
O6. O6. All right. Now, you see, like what I just said right now, that marriage door, they don't close it. Now, let's say the glucose has moved into the cell here. The glucose is inside the cell. Okay, my glucose is here. Okay, so it's 6 carbon. So let me draw my 6 carbon. Beautiful carbon here. Beautiful carbon. 6 carbon, right? 6 carbon molecule like that. In the statement I made. Now, this glucose moved from here. 1, 2, 3, 4, 5, 6 carbon. Pass through this cell and then enter cell wall, cell membrane, and enter the cell. Now, the way the glucose entered the cell, the glucose can come back as it went. Please, do you get my argument here? Do you understand this first statement that I've made? The way glucose entered the cell, the glucose can also exit the cell. The way you entered, you can come out the same way you entered. Like you get into somebody's house and then we'll show you the door. That's, my friend, the way you came, just get back like that, just the way you came. Okay? So, how would this glucose stay? How do you, the glucose, once it is in the form of glucose, 6 carbon like that, the way it went in like that, it can come back the way. Why? Because that door for that glucose is not what? It's not closed. Okay? Just like marriage, it's not closed. Okay? So the way glucose went in, the glucose could come back. So what should we do to prevent the glucose from coming back? Then we'll break, ATP will come. We'll break 1 ATP, All right. You see, remember when we break ATP down, we'll get what? We'll get inorganic phosphate and what? And ADP. Like that. Okay. And this phosphate here will go and attach to here. So let me give it a box. So this is my phosphate here. We'll get attached. Let me even change the color so that it will be clearer. Okay, so let me change this one, please. We'll be breaking very soon. Okay, great. So now this ATP here has broken down into this. Let me increase the thickness a little bit. Okay, let me increase the thickness a little bit. Now has broken down into this plus this. Okay, ADP, like that. Then this phosphate here will come here, will come and add to this. Now, the moment we add the phosphate to the glucose, that phosphate will pin the glucose down, as some ladies do. All right?
Just like pinning them with pregnancy, pin them down.
Exactly. So no wonder the phosphate also start with P. You get it? So they will pin you down. Once they pin you down, the glucose can't leave the way it came. So the first step of glycolysis is called what? The first step of glycolysis, the first step of glycolysis is termed as what? The first step of glycolysis, okay, this is the first, it's called what? We call it phosphorylation of glucose. Okay, and this phosphorylation of glucose to form what? To form glucose 6-phosphate. Okay, so they will add the glucose, the phosphate to the 6th carbon, all right, to form what? Glucose, okay, glucose 6 what? Phosphate. So this one, so they've pinned this thing down, and the enzyme for that is called hexokinase. Okay, the enzyme that will catalyze the process is called hexokinase. So the first step in glycolysis is this, is the phosphorylation of what? Of glucose. We do this, so we need to break ATP down so that the phosphate will pin the glucose down in the what? In the cytoplasm, so that it wouldn't go back the way it came. So the first step of glycolysis is the what? Is the phosphorylation of glucose. Okay, that they will break glucose down so that they will use the phosphate to pin the what? To pin down. I said they will break what? ATP down, and then they will use it to what? To pin the what? The glucose, so that the glucose can't exit the cell the way it came. And the enzyme for that process is called what? Hexokinase. So the hexokinase is the one that is going to add the what? The phosphate to the what? To the glucose, so that the glucose can't exit the cell the way it came. So the first step of glycolysis is what? Phosphorylation of glucose. That is why we need a what? Another ATP will come in, but I think I've gone enough now on this one. Okay, so let me just give you a little bit of break. Ajaman, how many minutes break do you need? Okay, let's say we are coming back
10 o'clock. 10 o'clock.
Okay.
Yes,
right,
please.
great.
So please,
All right.
colleagues, 10 is 10. Oh. Victor, go and get a kinky fast. 10 is 10.
10 is
Maybe
10.
10 is too much. I can even use only 15 minutes to finish that kinky.
Okay. All right.
Hey.
But we know our
Sir,
people.
we have some strong jaws.
All right. Okay. So folks, please, we are coming back at 10 o'clock. Okay. all right, it is getting interesting. If we're able to finish even glycolysis and then the Krebs cycle, that's the glycolysis and within, because that's the greater part. Once we're able to finish the cellular respiration today, then the following when we meet again, okay, when we meet again, we're only talking about what? Photosynthesis. Okay, we need to take our time to.
Doc, this is the last. Today is the last meeting. Oh.
That's a technically today is our last meeting, but I don't want to make.
Frank, we are working on it. Don't worry. We are working on it. Say, don't worry.
We don't want to make today just the last. If not, I'll rush it and you will not understand. But you can see that I'm making a lot of notes. Okay, we are going because some of you are going to be teachers of biology, and some of you are going for masters and PhD. So I don't want to not get the concept. So I have to take my time and so that you grab the concept.
Please, Doc, may God bless you for that. You do all.
You are putting the tutorials.
Oh yeah, that track. No, the objects will come. I've given the track questions here. Okay, all right. So we'll go step by step. Okay, so that you understand it. Okay, all right. I want you to go to SS class and you go without book, like the way I've opened a page like that, and you are killing it. And then the teacher just you kill it for me. And then all right. Okay, please do that. Okay, so please let's give ourselves a little bit of break. And then we'll come back exactly 10 o'clock and then we'll wrap it up. Okay. All right. Thank you. Okay. Strong as I am, I will always worship you. Hey, folks are still not back from the break. They should let us hurry up so that we can nail this quickly.
So students and their break, they will never come.
Hey, my people. We are coming.
So I get a cane when they come. Sound get one lash.
No, what they do is when they come and the place is quiet, they exit again. So
Okay, but I can see them coming. So yeah, so I think we've gotten maybe one more person and then we'll get started. Okay, I think we have to start now, so we can't be waiting forever. Okay, so in glycolysis, they are saying that the first step of what? Of glycolysis is what? Phosphorylation of glucose. Okay, phosphorylation of glucose to produce glucose 6-phosphate. All right, and glucose 6-phosphate can't leave the cell just the way it came. So glucose 6-phosphate is like pinning down. that the molecule here, this is pin, okay? This is like pinning down. And with the pinning down, you can't go back the way you came. So the glucose here is now converted to glucose 6 phosphate. And glucose 6 phosphate, okay, cannot leave the cell the way it came. And this process is catalyzed by what? An enzyme called hexokinase. This process is catalyzed by an enzyme called hexokinase. Okay? This process is catalyzed by an enzyme called hexokinase. So this is the first step of glycolysis, which is very, very, very important step. Okay? So this is what one of the ATP is used for. Okay? So another ATP will come in later. So this is glucose down. We use one ATP to pin it down. All right? We use one ATP to pin it down. And then the other one, okay, the other one will come in later. So the first step is very, very important. It's the phosphorylation of what? Glucose. Okay? Phosphorylation of glucose. All right? So that being catalyzed by an enzyme we call what? Hexokinase to form what? Glucose 6 phosphate. Okay? Beautiful. Okay. So now glycolysis, then so let's go to the second part. The second part, yes, here. Oh, not this one. Okay, now then we have the word, we have the pyruvate. So this pyruvate, so now the first step is what? Splitting the glucose. Okay, there are a lot of steps in there. Okay, but I've shown you why the ATP is what? The ATP addition is necessary for the what? For the work. Okay, so what happens is that now we have the word, we have the so the 2 pyruvate here. Okay, the 2 pyruvate here. So the 2 pyruvate, there are 3 carbon molecules. Remember, glucose is 6 carbon molecule. All right, glucose is 6 carbon molecule, and pyruvate is what? 3 carbon molecule. Pyruvate is what? 3 carbon molecule. All right, 3 carbon molecule. Okay, pyruvate is 3 carbon molecule. Okay, so with a 3 carbon molecule, okay, so that which means that the 6 carbon molecule here will be broken down into what? Into 3 carbon molecule, 2 of it. Okay, and this one here we call it what? We call it nicotinamide adenine dinucleotide. Are you with me? We call it what? We call it nicotinamide what? So we have this is, you see here, because of the hydrogen, we say this has been reduced. All right, this one has been what? Reduced because of the hydrogen. Okay, because of the hydrogen, we say it's what? We say that, okay, it is what? Nicotinamide what? The full meaning. The full meaning is what? Nicotinamide, okay, adenine, okay, dinucleotide, okay, and here, so this is hydrogen, okay, all right. Or if you don't want to say it like that, you say that reduced. Please, do you understand what I'm saying here? When you use the word reduced means what? Reduced means hydrogen has been what? Added. Please, are you with me? So you can say that it is what? Nicotinamide. So let me enter. So let me copy this thing here. Okay, like that. Okay. Okay, like that. So let me take them one by one here. So we have a word. We have my pyruvate, okay, being what? 3 carbon, 3 molecule, 3 carbon, okay, 3 carbon molecule. All right. And then the ATP is the word. That's the energy. And then we have the word. We have the ANDP. Okay, the ANDH. Sorry, NADH. So you can say it is reduced word. Nicotinamide. Nicotinamide, adenine, dinucleotide, nucleotide. Okay, when you put the reduced here, it means hydrogen has been added. If you don't want to put it that way, you can say this hydrogen. Are you there? Like that. But when you put reduced here, everybody knows that then there is what? There is hydrogen added at the back here. That's reduced, addition of hydrogen. So nicotinamide, adenine, dinucleotide, hydrogen. Or reduced nicotinamide, adenine, dinucleotide, hydrogen. Are you there? Okay, good. So this is and this particular substance is very, very important. This substance here, okay, it is what? It is the NADH. We refer to them as what? Electron carriers. Okay, we refer to them as what? Electron carriers. So they carry what? Electrons. Okay, they carry high energy. High energy electron. Okay, so they are what? Electron carriers. Okay, they're electron carriers. Okay, so they carry high energy electron. Okay, good. So recap. Glycolysis. Let me put glycolysis here. Is a splitting of glucose into 2 pyruvate, okay, molecules for ATP and 2 NADH. But during the glycolysis, 2 ATP are used. And because 2 ATP are used, the net ATP of glycolysis is 2. And then the reason the ATP is needed is because the first step of glycolysis, which we call the phosphorylation of what? The first step of glycolysis, which we refer to it as what? Phosphorylation of glucose, the addition of the phosphate. This phosphorylation of glucose is what will pin the glucose down by changing the glucose into glucose 6-phosphate using an enzyme, the enzyme called what? Using an enzyme called what? Hexokinase. Okay, so this is the enzyme hexokinase that is going to catalyze the what? The conversion of the what? Of the glucose into glucose 6-phosphate. Okay, so that the glucose cannot what? Exit the same way it came. All right, so that is how come the ATP is actually one of the reasons ATP is required for glycolysis to take place. Okay, all right, good. So quick on this side. So we know glycolysis, okay, splitting of glucose. Okay, the first step, okay, of what? So in cellular respiration, glycolysis is actually the first step, okay? And one quick thing here that is not here is that glycolysis is actually exhibited by almost all living things. So almost all living organisms, okay, almost all living organisms would undergo glycolysis. Almost all living organisms undergo glycolysis. Almost all living organisms undergo glycolysis. Okay. Almost all living organisms undergo glycolysis. Okay. Almost all living organisms undergo glycolysis. Okay. So it is an ancient process. Okay. It is an ancient process. It is and word ancient word process. Okay, it is an ancient word process because almost all living organisms undergo glycolysis. So it is an word ancient process. Okay, any headache so far? Okay, any headache so far? Any headache so far? Any headache so far? Okay, good. All right. So glycolysis is very, very, very important. All right. It's one of the main steps. ATP is produced. Electron carrier is also produced. And then what? So it is parts of the word, from parts of the word, the aerobic. All right. And then so here for ATP, both all these ones are what? The NADH is actually required for other processes as well. And then production of pyruvate is used in where? The next phase, which we're going to talk about it. Okay, all right. I don't want to go into much detail of this one. The most important what I've highlighted here, I'll go into detail of it in the next step. Okay, good. So glycolysis, the three main stages of cellular respiration. Glycolysis is number one, and it involves the conversion, the splitting of glucose into 2-pyruvate with a net release of 2 ATP and 2 NADH. All right, it occurs in the cytosol, and then it's exhibited by almost all living things, almost all living organisms undergo that. Almost all living organisms undergo that. Okay, so almost all
What
living organisms.
the cytoplasm and the cytosol?
The word. Hello, come again.
The cytoplasm and the cytosol.
Yeah, so well, we have the it's actually the same thing though. The cytosol is actually part of the cytoplasm. Okay, so you see that the outer part of the cytoplasm is cut. The outer part of the cytoplasm, where the cell membrane, where it is close to the cell membrane. Okay, where it is close to the cell membrane. Okay, where it is close to the cell membrane, that part's a little bit hard. All right, but the inner part, the within the cytoplasm itself, okay, that's the liquid component of the cytoplasm. That's where we have the cytosol. Okay, all right. That is why we use the term. I use the term interchangeably. Are you there? So the outer part is a little bit hard. Hello, come again.
So when you were explaining, you said the dicotinine adenine the NADP.
Yes. NADP. H is the H.
Yes.
H.
Okay.
Yes. So
what is that when hydrogen is added? That is when we have the
so this is one. So this one, because of the hydrogen is here. Look at that. There's hydrogen here. Have you seen it? We have a molecule like this. There's a molecule like this, NAD plus, like that. This is nicotinamide adenine dinucleotide. Are you there? I see this is plus. So this one is more or less plus. So it's like iron, right? Now, when it reacts with oxygen, it has been reduced. Remember, addition of hydrogen is reduction. Are you there?
So, yes, please. So
Go
what
ahead.
is the function of the hydrogen being added to the...
That is how this one is the molecule. The
That
function
is
of the
how it is.
hydrogen. Yes. So this one here, in the course of the reaction, this is what will be produced in the reaction. This molecule will be produced there. But what you are saying, just like the way we have something like this. We have ADP, and then they will add P to it. Okay, they will add P to it, and it become ATP. It's the same way. So in the cells, we have NAD in the cells already in the cytoplasm. You understand? But during glycolysis, okay, this molecule will be there. This molecule is required in the glycolysis. This molecule is there already. This molecule is required for glycolysis to occur. Are you there?
Yes.
So during glycolysis, the hydrogen will be where? Hydrogen will be added to this. Okay. So here, without a hydrogen, I can just pronounce it nicotinamide adenine dinucleotide. I can say that. But when the hydrogen is there, I can say reduced nicotinamide adenosine dinucleotide, because when hydrogen is added to a molecule, it is what? When hydrogen is added to a molecule, it say that reduction has taken place from the radius reaction I explained.
Yes.
Okay. Yes. So during the reaction, this particular substance is available. NAD plus is already in the cytoplasm.
In the cytoplasm. Okay.
Yes, it is there. Okay. So if we are going to write full version of what? Glycolysis. Let's say if I want to write full version of glycolysis, I write it this way. If I'm going to write a full version of glycolysis, okay, let me go there. I'll write it this way. I write it. Full version of I'll write it with glucose, okay, plus what? NAD plus A 2 ATP, giving me 2 pyruvate plus 2 NADH, okay, plus 4 ATP. All right. And remember, when you break ATP down, what do you produce? You produce ADP. So you broke 2 ATP. So if I want to write detailed glycolysis, this is how the overall formula will be like. So this one, okay, let me bring another plus. So this substance is already in the cytoplasm. We take our glucose there. We add our ATP. We get 2 pyruvate. This substance during the glycolysis, this one will be reduced. Okay, will be reduced, and then to become this. And so what is here? We call it electron carrier, high energy electron carrier. And then we require ATP. We know why we need the ATP. We produce 4 ATPs. Okay, And if I write that's the one there, which means that I have to write, because we need 4 ATP here. So the total one, I have to even add what is here. I have to write 4 ADP here. Because the 4 ADP here is what is going to become the 4 ATP. Have you seen it? So if we are to write the detailed reaction, we'll write, okay, usually we don't indicate all the, we just indicate the important energy stuff, okay? All right. So we indicate the important energy stuff, okay? So 2 pyruvate, 2 that, and then the word, and then the 2 NADH, all right? Okay, so that's the ones that we normally indicate. Okay, all right, great. So glucose plus 2 NADH. Glucose plus NADH. NADH, okay. 2 plus the glucose here plus 2 NAD, okay, plus what? Plus 2 ATP. Okay. Plus 2 ATP. Okay, plus so that will give us that, okay, and then we get my what, 2 pyruvate, okay, and then we can get 2 NADH plus 4 ATP. So this is the overall reaction that we are going to get. And as I said, what is here is an electron carrier, and this is the energy here, and so we have net energy of what, 2 ATP. Please, are we clear with that?
Yes,
are we
please.
All right, thank you.
So,
Please, Doc, one more time.
so Doc, my problem also is that so the NAD, its
Yes.
main function is to help the process of glycolysis in the
Exactly.
cell.
Okay, you see, we said that even though glycolysis, oxygen is not there. Listen, even though glycolysis, it doesn't involve oxygen, but it is still undergoing oxidation. The glucose is still undergoing oxidation. And when you are undergoing oxidation, you see, there are the definition that I gave earlier on. Oxidation can be defined as removal of what? Of hydrogen.
Hydrogen, yes, hydrogen.
Do you understand that?
Addition of
So
oxygen.
when we remove the hydrogen from glucose, we should get somebody to pick the hydrogen. Are you with me?
Yes.
Okay. So still, even though oxygen is not here, the glucose is undergoing what? Glycol is undergoing oxidation. And as it's undergoing oxidation, when you remove the hydrogen, there should be somebody there that will take the hydrogen. For instance, when money comes to you and you buy something, how will the money disappear? The money will not disappear into the tin air. The money will move from you to another person. Is that not it? Let's say you've been paid in a month. You've been paid 5,000 in a month. Now the 5,000 is with you. How will the 5,000 go? You buy stuff. So at that same time that the money is going, the money will not just go into the thin air. It will go to somebody. Okay, so somebody have to pick it. So let's take it this way: the Ajiman is here. Now, Ajiman is undergoing oxidation. Oxidation can be defined as addition of oxygen or removal of what? Hydrogen. So in the case of glycolysis is still undergoing oxidation, but just that oxygen is not involved. It is involving what? The removal of what? Removal of hydrogen. So that removal of hydrogen, somebody should be there to take the hydrogen. And that person is no other than nicotinamide adenine dinucleotide. You can see it is NAD plus. Okay, so it should be there. So those ones, they're already in the cytoplasm. All right, so they are supposed to be there to take the hydrogen from the word after the split of that, the splitting of the glucose, which is also a type of what? That's the oxidation reaction, which will involve removal of hydrogen. So the hydrogen should be there. Okay, the NADH, okay, must be there to do what? To accepts the word to take the hydrogen that will be produced. And so you can see that because hydrogen has been removed from oxygen, we will say that glucose, because hydrogen has been removed from what? From glucose, we will say that glucose has been what? Oxidized. And because hydrogen has been added to NADH, okay, plus, we will say that NAD plus has been what? Reduced to become NADH. Are you with me? Hello? My wonderful people.
We are with you.
Hello, we are with you.
Okay, good. So, good. This process here will all occur in the cytoplasm. Okay, so what is here is occurring in the cytoplasm. So before the conversion of before the glucose will be split into pyruvate to 2-NADH and then what, 4-ATP, what we need is that we need NADH to be there. Somebody should be there to accept. Okay, it's just like Ajingo has finally seen some fine chick in Tema. Okay, and want to this fine chick has succeeded to pin Ajingo down. What is going to happen now when Ajingo goes to see the family and gives the money as the bridal whatever? There should be somebody there to receive it. Okay, so that person who is going to receive it is the NADH. So when they give out the hydrogen, okay, somebody should be there to receive the hydrogen. And who is there to receive it? It's NADH plus. They are there to receive the what? The hydrogen. Okay, and when they receive the hydrogen, it means that they have been reduced. Okay, all right. So removal of hydrogen means you've been oxidized. When you receive the hydrogen, you've been reduced. Please, are you with me? So what I've written here is the detailed stuff of glycolysis. Okay, that before glycolysis is going to occur, we need glucose, we need NAD plus, and we need ATP. Okay, for glycolysis to occur. At the end of the glycolysis, you get 2 pyruvate. The pyruvate is what? 3 carbon what? It's a 3 carbon molecule. It's a 3 carbon molecule. All right, it's a 3 carbon molecule. All right, it's a 3 carbon molecule. Okay, and then remember this one is going to accept the hydrogen from the glucose, so it will be what? It will be reduced. So that when you say, if I'm pronouncing this, I will say that it's a reduced NAD. Okay, reduced nicotinamide adenine dinucleotide. Or I will say that nicotinamide adenine dinucleotide hydrogen. Okay, because of the hydrogen there, that's why it's reduced. Okay, then 4-ATP produced. So it's very, very simple. All right, good. So this is that for. So now a little bit much detail of glycolysis. So like the substrate for glycolysis. We all know glucose is the almighty substrate for glycolysis. Okay, we have glucose is the almighty substrate for glycolysis. So like before glycolysis is going to occur, glycolysis will be what? We need glucose for glycolysis to occur. All right, good. And sometimes depending in animal cell like this, okay, in animal cell like that, glycolysis can be what? Glycolysis can the glucose can be converted into glycogen. Okay, all right, and then so if let's say glucose is not there, they can convert the glycogen into glucose first before the glycolysis will occur. But occasionally we have some organisms that let's say they are taking in fat. Okay, do you understand that there are some organisms that they are not really taking glucose directly? They take in, let's say, fat. All right, fat. Okay, they take in those fatty acids, like let's say a lion. If I take something like a lion doesn't feed on plant. Okay, so lion is going to consume these animals, their fat and everything is going to be in this lion. Now what is going to happen is that, okay, apart from glucose, other glycolysis can occur, okay, without using glucose as its substrate. Okay, the substrate is what is going to work upon, like what the starting point. Okay, now there are instances whereby glycolysis will okay, but they will use what? They will use something like glycerol. Okay, you understand? So we have but the main one is the glucose. The main one is the glucose. But if glucose is not there, okay, if glucose is not there, cells could use fat. They'll convert the fat into glycerol, all right, and then they will use it to generate the what? The pyruvate. Okay, and occasionally, even protein, they can convert the protein into what? Into certain amino acids, all right, and then they will use the amino acid for glycolysis. Okay, so to go through a number of metabolic processes, and then glycolysis will occur. But the main glycolytic pathway, has what? Glucose as its substrate. That is, the glucose is the one that is going to act upon. But for any reason, if glucose is not available, the glycolysis is not going to stop. Because if glycolysis stops, that's a big trouble. Because the glycolysis is the first stage of the what? Of cellular respiration. Is the first what? Stage. The first phase of what? Cellular respiration. Okay? So if there is no glucose, that it's not going to stop. All right? What is going to happen is that other substances, fat could be used, and then some proteins could be used, okay, for the glycolysis to continue. Because the moment you stop, it means that ATP will not be produced. So if there's no if there's no glucose at all, still the glycolytic pathway will use other substrates, and the other substrates can use is the fat. Okay, they will just change the fat into glycerol, and then they will use the glycerol as a substrate, okay, for what? For conversion into the pyruvate. And even occasionally, okay, they can use what? Protein, they'll convert protein to some amino acids, okay, like alanine, and then the glycolysis will continue, okay? But the primary mechanism is glucose. Glucose is the main thing that is going to happen. That's the primary process, okay? The primary substrate for glycolysis is glucose, okay? All right. But even in case glucose is not available, they can rely on what? They can rely on fat, okay, or rely on protein, okay, whereby they will change the fat to glycerol, or they will change the protein to amino acid, and then before it enters into the what? The glycolytic pathway, okay. All right, good. Okay, now this is actually what I've just talked about earlier on, okay. In your case, we will not go to, if let's say if I were to be teaching the undergrad, this is what they are supposed to know. They are supposed to know all the 10 steps of glycolysis. From here, this one here, literally, okay, they are supposed to know all the 10 steps, and they are supposed to know all the 10 enzymes, okay. All right, so they are supposed to know this, okay. They are supposed to know all the 10 steps of glycolysis. But in your case, okay, we'll just take just few of the steps, okay. All right, good. We'll just take few of the steps, okay, and then okay, but ideally, okay, this is all the 10 steps, okay. That's what we require, okay. But I will highlight, I brought this one here so that I will highlight some few things in there. Now, if you take my glycolysis here, you can see the glucose is here. You see, as I just said earlier on, the glucose is, I'm using the glucose as my substrate, and this glucose as my substrate is what? It's a 6-carbon what? It's a 6-carbon sugar. And then the first step of what? The first step of glycolysis is to pin the glucose down. All right. And that first step, as I said, is what? Is the what? Is a phosphorylation of what? Of glucose. So that I have to just make sure the glucose is pinned down. Like Ajaman said it, like sometimes your side chick may pin you down with pregnancy. So that pregnancy is a phosphorylation of glucose. Okay, so now you can't just run away. The hit and run method wouldn't work this time. Why? Because you've been pinned down. Okay, so the first step of what? If you look at this, if you look at my screen here, okay, if you look at my screen here, you can see that the first step here, the first step on my screen is the what? Is the phosphorylation of what? Of the glucose. Okay, if you look at it, the first step on my screen here like that is the glucose, and then they what? See, they break. If you look at the top here, okay, okay, let me zoom my screen. Let me zoom my screen like that. Okay, great. Okay, if you look at the top here, you see, look at the first step. So the first step of what? The first step of glycolysis is the glucose here change into what? Glucose 6-phosphate. Okay, so that is where the one of the phosphate here, okay, one of the phosphate will be added at the what? At the 6th end here. All right, so one of the phosphate will be added at the 6th, okay, the end here. That's where you get a glucose 6-phosphate. And then if you look here, the second step, you see that the second step, so when we change glucose 6-phosphate, then the second step to undergo a certain process, we call it isomerization. Isomerization, I think, I'm sure you in your chemistry class, you know something called isomerism. So isomerism is that when you say you are isomer of something, it means that you have the same structural formula, okay, in terms of your the same molecular formula, okay, but your structure formula, your structure formula are different, okay. So you have the same molecular formula, but your structural arrangements are different, okay. So maybe 2 or more molecules having the same word, the same molecular formula, but their structural formula are different. Okay, good. So here, if you look at the first step in glycolysis, that is the phosphorylation of glucose. So they pin this one down, changing into a glucose 6-phosphate, and then this glucose 6-phosphate, okay, is catalyzed by what? Remember, it's catalyzed by the hexokinase. This glucose 6-phosphate will undergo something we call isomerization. Isomerization is just simply rearrangement, okay, of the what? Of the glucose 6-phosphate to become something we call fructose 6-phosphate. Okay. Now, when this fructose 6-phosphate is made, that is when it's going to do what? It's going to the fructose 6-phosphate is going to do what? It's going to change the they will add another phosphate to it. Okay, you add another phosphate to it. Now, where you need to, the way to keep this one in your head is that any reaction that involves phosphate, you will see the name has kinase at the end of it. That's how you memorize it. Any reaction, okay, that you see kinase in front of the enzyme, okay, the moment you see kinase, that one tells you that it means that that reaction has phosphate involved in it. Okay, so when you say hexokinase, phosphofructokinase, okay, anytime you see kinase in there, it means that there is what? There is phosphate in there. So here, what you are going to do is that turn this one, the ATP, all right, breaking down to pin this one down. Then it will undergo isomerization, okay, that the glucose 6-phosphate will become fructose 6-phosphate. And when it becomes fructose 6-phosphate, okay, then it becomes what? It becomes then it will undergo something we call another ATP will be what? Will be broken down. So this is how the 2 ATPs are used. Okay, I don't want to go through all the details with you, but I just want to show you how the 2 ATPs are used. Okay, so you see the first one is to pin it down so you don't go back the way you came. And then the second one, the second step, you go isomerization. And then the third step, then they will do what? This fructose 6-phosphate, they will add another phosphate to it. And when they add another phosphate to it, what are we going to get? You are going to get what? You are going to get fructose, what? That's a phosphofructokinase here. This is the enzyme here that will ensure addition of what? Another ATP to this. Ensure another ATP to this. And change it to what? Phosphor-1,6. So the phosphor-1,6 what? That's a phosphor-1,6 what? Bisphosphate. So remember, bi means 2, right? Bi means two. So here, so now what I'm showing you here, this what is in this side, okay? This side here is what? What I'm showing you here is a what? Is the fructose 1,6, okay, bisphosphate. This what is here? Fructose 1,6 bisphosphate here. All right. Okay. Let me take it one more time. First of all, I get my glucose. The glucose comes into the cell. It can go back just the way it came. So what do? We want the glucose to stay. So we want to pin it down. So we pin the glucose down by adding what? Phosphate to the what? To the 6th carbon, so that we get glucose 6-phosphate. So when it happens like that, we say the glucose has been phosphorylated. So after the glucose has been phosphorylated, now the glucose will not go back just the way it came. Okay. So what is the next thing going to do? So as the glucose is not going to go back the way it came, now then it will undergo something we call isomerization. Okay. Isomerization is the glucose 6-phosphate is going to turn into something we call fructose 6-phosphate. And when it turns to fructose 6-phosphate, okay, then another ATP will be broken down again, and then add it to the what? Add it to the end of the fructose 6-phosphate. Okay, so this diagram you are seeing on my screen, this second diagram you are seeing on my screen, I say phosphate with the glucose with 2 phosphate at the end is what? Is fructose what? Fructose 1,6. You see, one is attached to carbon 1, another one is attached to carbon 6. So 1,6 bisphosphate. Okay. And what is the name of the enzyme? Well, I don't expect you to get the enzyme, but the enzyme is going to be what? The enzyme will be the what? That's the fructose what? That's the phosphofructokinase. All right. So anytime they add, don't forget that the enzyme, it happens that you've not even heard of the enzyme before. The moment you hear kinase at the back of it, it means that there is phosphate involved. So phosphate is what is involved. Okay. Now, okay, I just want to use this detailed diagram to explain. You don't necessarily need to get the detailed diagram, but some key things so that we will see where the NADH came in, where this one came in, and stuff like that. Okay, so that we will appreciate it. Okay, good. So this one. So first of all, we pin the glucose down. The glucose undergo isomerization. Okay, to get fructose 6-phosphate, then we break another ATP to get 1,2 fructose 1,6 bisphosphate. Okay, then afterward, what are we going to do? This one is highly unstable. Fructose 6-phosphate is unstable. So what is it going to do? It will break down. All right, it will break down. When we break down, we'll get one molecule. We'll get what? Hydroxyacetone. Okay, hydroxyacetone phosphate and glycolaldehyde. All these are 3 carbon molecule. But do you know something? This hydroxyacetone can be converted. In fact, it will be converted. The two of them, this molecule here, okay, can easily quickly be converted into what? Into glycolaldehyde, which is a 3 carbon molecule. So let's see something here. So what you are seeing here, okay, what you are seeing here, you can see it is glycolaldehyde 3-phosphate. Okay, when we break this one down, okay, so let me see. When we go our detailed one here, now we get our fructose 6-phosphate, okay, which is 6 carbon with what? With phosphate at the end. All right. Then what is going to happen? What is going to happen is that there is an enzyme, we call it that's the aldolase. Okay, this aldolase, what is going to do is that to break this. So the aldolase here, aldolase will come and break this one here. This particular this 6 carbon here is the work of aldolase to we call it cleavage. Okay, so to cleave here, all right, and break this one into 2. But when it breaks this one into 2, what are we going to get? We will get something, 2 substances. We get what? We get We get dihydroxyacetone phosphate. and then glycoaldehyde 3-phosphate. But the diacetone, this dihydroxyacetone
Anita, please, your mic is on.
can be what? Can easily be converted into the glycoaldehyde 3-phosphate. So because of that, we don't even really talk much about the word. That's a dihydroxyacetone, because the moment it is formed, it is converted to the word, to the glycoaldehyde 3-phosphate. Okay, so what you can see here, these two molecules that you can see here, they are what? They are glycoaldehyde 3-phosphate. Okay, but which enzyme is going to break this into two? It is what? That's a aldolase. Okay, the aldolase will break this. So what I have here, the two with this 6-carbon molecule with 2 phosphate at the end, we are referring to what? We are talking about that's a fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. All right, fructose 1,6 what? Bisphosphate. That is the word. That is the one that is the one here. Then this one is highly unstable, so it must break. So we break it down. And who is going to do the cleavage? Who is going to break this 6-carbon molecule into 3-carbon molecule? Then an enzyme called what? Aldolase will come. We'll break it here. Boom. We'll break it here like that. Okay. We'll take this one here. Okay. We'll break this one here. Oh. We'll break this one like that. Okay. When we break this one, we get the what? We get the dihydroxyacetone phosphate. And then we get what? We get the glycoaldehyde what? Glycoaldehyde 3-phosphate. But the moment we get the dihydroxy, the dihydroxy what? Dihydroxyacetone phosphate quickly. So that's the DHAP. Quickly to be converted into what? Into glycoaldehyde phosphate. So this molecule you see here is the what? Is the glycoaldehyde 3-phosphate. This molecule you see here, glycoaldehyde 3-phosphate. Okay, but it started as glucose. Okay, it was pinned down by hexokinase. Okay, by breaking what? By breaking ATP in the cell, ATP in the cell, and you add what? 1P here, 1P at the end to pin this molecule down. Then the molecule, the glucose 6-phosphate, we undergo isomerization, rearrangement, isomerization to give rise to something we call what? Fructose what? Fructose 6-phosphate. Okay, in this fructose 6-phosphate, another ATP will be broken down, and then they will add what? They will add another phosphate to it, like that. So one phosphate will come here. When they add another phosphate to it, we'll get what? We'll end up getting what? We'll end up getting, that is the fructose 1,6-bisphosphate. Because 1 is at the end here, there's 1, 2 at the end, making it the bi. That's where we have the what? The bisphosphate. Then at the end there, then there's an enzyme called what? Aldolase. Okay, this enzyme here, the aldolase here, aldolase will come. The aldolase will come and break this 1,6, okay, a fructose 1,6-bisphosphate into what? Into DHAP, dihydroxy what? Acetone phosphate, into what? Into glycoaldehyde 3-phosphate. All right, good. But as I said here, the moment you get it, it will quickly be converted into that. Okay, even though an enzyme will be involved, you can see that the enzyme is what? Isomerase at the end. So it's basically rearrangement. So you can see trials what? Phosphate isomerase. So when you see isomerase, it's purely rearrangement, just like the way we have the phosphoglucose isomerase. It is purely rearrangement. Then when we get here, so this on this side here, which you see on the slide that I gave you previously, if you see this one here, so here the three, this was split into two. And so at the end of the day, we are going to have what? Glycoaldehyde 3-phosphate here. Okay. And when we get our glycoaldehyde 3-phosphate, this glycoaldehyde 3-phosphate will then be converted into what? Pyruvate. So let's see how. Okay, good. So if you look at my screen, let me zoom it up a little bit. Good. So from here, now look at that. You see, from the glycoaldehyde 3, that is where the NADH plus comes into being. You see, when NAD plus comes into being, you can see that it has been reduced. You can see there's reduction there. Okay, there is what? Reduction. So this, they've removed what? Hydrogen from it. Okay, you can see there is removal of what? Of hydrogen. They are taking hydrogen, okay, so and then some of the hydrogen has been added to this particular molecule. All right, so you can see, have you seen it? So you can see that on the 6th phase, that is where this oxidation reaction is what? Occurring, whereby they will pick what? They will pick hydrogen, they will add hydrogen to this to become NADH. So it is that the 6th step. Okay, and then so when you get this what? When you get this hydrogen after the oxidation here, then we have the word the 1,3-bisphosphate word glycerate. And look at that. Why I'm showing this side, I want you to see how the ATP will be generated. You see, between on this 7th point here, you can see that 2-NADP has come into the picture. All right, 2-NADP has come into the picture, and then the 2-NADP, the 1,3-bisphosphate, okay, like that. All right, now each of them, now listen to me, each of them it is one, but because in the reaction you can see that, you can see here the 2 pyruvate molecules, okay, produced. All right, you can see that here at this step here. You can see at this step here, we are having 2 glycoaldehyde 3-phosphate. Okay, so we have 2 glycoaldehyde 3-phosphate, and each of them will go through that step. Each of them will go through that step. So that is why everything here is ×2. Okay, but here it's only one that will be produced. But because 2 of the word 2 glycoaldehyde 3-phosphate has been produced, that is why everything here is ×2. Please, do you get the argument? So here, the process here, at the end of this, you can see the 2 is here. Just like here, you can see 2 glycoaldehyde 3-phosphate produced. Okay? And when this one is produced, what is going to happen? What is going to happen is that, so this is produced, this is produced. All right, and each of them will go all the way down here to the pyruvate. This one will go all the way down here. Okay, let me pick a marker here. All right, so this one will go all the way down. Okay, all the way down here to the what? To the pyruvate, like that. This one will go all the way down to the pyruvate, like that. All the way down to the pyruvate. Okay, all the way down to the pyruvate. So it's very, very, very simple. Okay, all the way down to the pyruvate. So because of that, that's why everything is ×2 here. Everything we just indicated 1, and everything right ×2. So here, now this glycoaldehyde 3-phosphate, each of them. So here there's going to be what? There's going to be oxidation, and this oxidation is basically removal of what? Hydrogens. Okay, and when they remove the hydrogen, we are going to form what? That's a 1,3-bisphosphate glycerate. Okay, all right, 1,3-bisphosphate glycerate, and you can see that even 2 Remember, you can see there even you can see two phosphate, inorganic word phosphate are even here. Okay, so they will add the word 1,2 bisphosphate. Okay, and then the next step, what is going to happen after this oxidation? The next step, this 1,3 bisphosphoglycerate will be what? Will be able to what? Give 1 phosphate, okay, to release ATP. But as I said, the process are 2. That's why it has been written times 2 here, because the what? The glyceraldehyde here, there are 2 of them. So each one of them is going to what? Is going to break the molecule down, okay? It's going to undergo this process, and then we'll break the what? The phosphate will be added to the ADP to generate ATP. Okay, so in glycolysis, all right, in glycolysis, the generation of the ATP occurs in the what? In the 3rd and what? The 10th step. Okay, so you can see, sorry, the 7th, the 7th and the 10th step. Okay, so one will be produced here, one will be produced here, but because the what? The glyceraldehyde 3-phosphate, there are 2 of them. Each of them will go through the process. So everything then will be what? Will be times what? 2. Everything there will be times 2. So times 2 here, times 2 there. Okay, all right. So times 2 here, okay, and times 2 there. So at the end of the day, you can see that we are getting 2 ATP here on the 7th step, and then 2 ATP on the 10th step before we produce the pyruvate. So at the end of the day, you can see from the top here, we spend 1 ATP here, 1 ATP here. That's 2 gone, and then we generated 2 ATP on the 7th and 2 ATP on the what? On the 10th step. Okay, and so we'll get 4. We'll produce 4 ATP, and then but we spent what? 2 ATP. How did we spend the first ATP? We used it to pin it down, and after that, after isomerization, we added the other one to the what? To the fructose, and then after adding it to the fructose will become what? Will become a little bit unstable, and then an enzyme will come, what? That's the aldolase will come, split it into 2, and when it splits it into 2, we get our beautiful glyceraldehyde 3-phosphate. Okay, and this glyceraldehyde 3-phosphate, okay, remember we are going to get what? We will get DHAP first, and by DHAP first, we will undergo isomerization and convert to the glyceraldehyde 3-phosphate. So all of them will have 2 glyceraldehyde 3-phosphate, and then it will continue. Okay, that's where the oxidation will take place with this one here. All right, adding the what? That is the taking away oxygen from hydrogen from it. Okay, taking away the hydrogen. Okay, we get a 1,3 bisphosphate, and then it is at the 7th step that ATP will produce, and then at the what? At the 10th step, that is where what? Another ATP will be what? Will be produced. Okay, good. You don't need to know this in detail, but I needed to talk about those ones so that you understand how we jump from here to here and how our 4 ATP came about. Okay, but what you need to know is that we started with 6. Okay, we started with what? 6 what? We started with 6 carbon molecule, which is the glucose. We pin it down with phosphate, which is the phosphorylation, using an enzyme called hexokinase. This one undergoes and form what? That's a 6 glucose 6-phosphate, which will undergo isomerization to produce what? That's a fructose 6-phosphate. And then another ATP will be broken down and then join the 1, so that we get what? We get fructose 1,6 bisphosphate. Okay, all right. And this one, an enzyme called aldolase, okay, will come and then break this molecule into 2. When it breaks the molecule into 2, we get 3 carbon molecules, which is what? Which is the dihydroxy hydroxy hydroxy dihydroxy acetate, okay, phosphate, all right, and glyceraldehyde 3-phosphate. But when you get a dihydroxy phosphate, so quickly the dihydroxy, okay, will quickly undergo what isomerization and change into what the glyceraldehyde 3-phosphate, and these two molecules will now continue all their way down to the what to form the pyruvate. So you can see that I have 2 times pyruvate, okay, on my screen. Okay, so that is why every step on what going here, everything here is what times 2 of everything throughout, times 2. Okay, and then on the 7th step, ATP is produced, and then on the what on the 10th step, another ATP is produced. And if you check the enzymes there, you can see, as I said, anywhere the enzyme phosphate is involved, you see kinase. You see here production of the ATP. You can see the pyruvate kinase as the enzyme. If you look at the 7th, okay, the conversion of the word of the 1,3-phosphoglycerate, okay, to 3-phosphoglycerate. You can see that word. You can see that we have the phosphoglycerate kinase. Okay, so even when you don't, so the moment you see kinase, it will tell you that phosphate was involved in the reaction. Okay, so here 2 of everything here, and that's how come. And then at the last one, that's a pyruvate will be converted into what? Pyruvate. Okay, and this will end the what? The glycolysis. Okay, good. So in glycolysis, what do we get? Okay, I think my last statement, you have another class, so I will end it here quickly. Yeah, hello, please.
Yeah, so please, earlier you stated that glycolysis involved the breaking down of glucose to release, to produce, pyruvate and 4-ATP and 2-NADH, if I'm right.
Yes, you're right.
Yeah, so, but in the slide, in the steps for glycolysis, I've noticed that we're having 2 pyruvates, 2-ATP, and 2-NADH.
No, you can see time. Everything is times 2. Look at that. Look, so here. Look at this one.
I mean downward here.
Downward here. Okay.
The product.
Down
here. The product of
here.
glycolysis.
Yes.
No, please.
So let's see. So
I mean the steps. Please, not here. The steps. The steps.
Yeah.
Yeah. So downwards. I mean the products. Where you having the product there?
Okay. So here, this ATP is net ATP. Net.
Oh,
this is net product. So here you can see that here. Look at the 2 ATP here. You can see the 7th step, because it is 1 ATP, but the steps, the products are 2. Remember what is here? Glyceraldehyde here going there. There are 2 of them, right? And each of them will go through this process. That's everything here is times 2. Have you seen it? That's a times 2. So here, ATP will produce here. Another 2 ATP will produce, so making it 4. Okay. So each of them is producing 2 ATP. Each of them. So in total, we have 4. But we have spent 2 here. All right. So this ATP here is net ATP, as it is being shown in this my last slide. You can see here net. Have you seen it? The net didn't come, but it is what? Net ATP. Are you there? So it is what? Net ATP. Because you spend what? We spent what? Spent 2 to get 4. Is that not it? Okay. So the net, so like the tax you paid is 2, and then your take-home salary is 4. So at the end of the day, you receive your gross salary is what? It's 4 ATP, and then your tax you pay is 2 ATP. So your take-home salary, which is the net, is the what? Is 2 ATP. Okay. So we have the what? We have the 2 NADH produced, okay, and 2 hydrogen ions there as well will be removed. Okay. All right. And then we have the what? We have the 2 pyruvate. Okay. So the most important take-home message here is that in glycolysis, we have 2 net ATP, okay, 2 NADH, and 2 pyruvate. All right. So that's the most important thing you need to know in glycolysis. Okay. I think you have the next class. So I will leave you here, okay, and provide you with this slide, okay, so that you can go over it, okay, in your spare time. And if you have any headache, feel free to give me a shout and I'll respond to you. Okay. I wish you all the best.
Hello, sir.
And the hello.
Please, last week's slide, we've not gotten it yet. Though we are able to.
Please, just something down, but you want the slide. Yes, I will do that.
Yeah.
Please, I'll get it to you.
Yes, please.
Okay. Once a lady has made the request, who am I? Not okay. Okay. I've been asking for it for a week. I didn't mind him, but once the request is coming from you, please, your wish is my command. I will surely do that.
Thank you, sir.
No, sir, what you said I didn't hear. I'm sorry. I am male voice. I am my ears can't hear things that okay. Right, I'm sorry. Ajiman, I can't hear what you are saying.
Hello, sir.
Ajiman, a lady is talking. Please let me listen to the lady first, please. Yes, madam.
Yes.
Yeah, please.
Sir, please. Can it be possible that we can meet before next week?
Yes, Fidelia,
Oh, yes.
He's not from this platform. Go to your platform. Go.
Mr. Ajiman.
Hey, Ajiman. Hey, Ajiman. Comport yourself. You are not romantic at all.
Okay.
Talk to a lady.
I mean,
Ajiman, you are one. You are one.
You were late. You've spoken about it already.
I'll
you see.
Yes, I'll remove
Thank you for removing me,
yes.
And that will even give me peace of mind to learn on my own because the course are so much. All right, sir.
So my lady, please. I think at the end of the class, Ajiman will lead you to discuss on the time that you want us to meet, okay? Because I really want to finish this one with you, okay? And that's why I've taken my time step by step. I'm not in haste. I just want you to understand it, okay? I'm not running with it, okay? I want you to understand it. So I think at the end of the class, they said it earlier on, okay? So they will discuss, and then they Ajima will let me know when it will be ideal. But the earlier, the better, so that it will give room for the slow learner. You know, right now the brain is not as sharp like as SS days or your university days. So you see, so we need to make sure we grab the thing early so that the slow learners will have ample time to revise. Okay, so my lady, please, Ajiman will communicate that one to you, and then we'll get that one sorted. Okay, thank you. Now, Ajiman, you can talk.
Thank you. Say bye bye. You can sleep. We'll continue. We'll get back.
All right. All right, great. So I'll provide the slides to you. Okay.
Okay, thank you, sir. Please, colleagues, at the beginning of the class, we discussed behind closed doors even before we came here. You know, doctor is such a busy man that he can call him from Monday to Friday. He'll reply all your messages from 10 p.m. So we agreed, partially we agreed that if things be okay, why don't we sacrifice tomorrow Sunday too? Then we meet him, finish with everything. Because we are left to talk about photosynthesis, go through the trial questions, and then the other ones that he will send. As he's saying, some are slow learners, some can just grab everything. The next week, next week is practicals week. After practical, the following week is quiz. After the quiz, then examination. So he's proposed that was behind closed door before even we came to the platform, that if we can sacrifice tomorrow to Sunday, so that we take us from 7 to our usual time, we are left to finish with Krebs cycle and then electron transport and then look at photosynthesis and any other thing. So he is suggesting that if we make time for him tomorrow morning, you meet it. I propose Sunday because from Monday to Friday he's quite busy. You call him and say, I'll get back to you, and that is the end. That is what he's proposing. If you all agree, tomorrow morning.
Hello.
Yes, Lord.
So, Adima,
No, it's okay.
please.
Wait,
That's all.
wait.
Yes,
Lord is talking. But let me listen to Lord first, please.
Please,
Lord.
I'm saying that tomorrow is okay. So I don't think we
Yes.
need
Let us all sacrifice our Sunday. For how long we have not been going to church, and God hasn't cancelled our name. We are still Christians. So
Hello, Adima.
let us agree that tomorrow. Yes, who
Hello,
is
Adima.
this? Benedicta.
It's Bernadette.
Yes.
Please, if
Wait,
possible,
Fidela, let me listen to someone.
let it be tomorrow, but why would I shouldn't be around 11 there? So that by that time, we are in the house.
Please, tomorrow.
No, please, let's come and so that we close early.
Sometimes in the afternoon, the networks are not that stable. If you have observed that, if you have lectures in the afternoon, we tend to have challenge with our network. So please, let us all. We've been sacrificing our Sunday since the beginning of this program.
Sunday morning, better.
Let's do it tomorrow morning and end it once and for all. Please.
Hello, Fuima.
Who that? Who is this?
Hello, Mr. Adman.
The course is a weekend course. So if you think you will not sacrifice it, before signing for it. So please, let's do it tomorrow morning.
Hello, please, my brother.
Tomorrow, let's do it.
Hello, Mr. Adman.
Yes, Julie.
Then please, if we do it tomorrow, sometimes you come and you'll be sitting down. Let's come early. Some of us will have programs to attend tomorrow. So if we come early, we can close early so that we all go for our various programs that we have. That is my plea because yes, please.
Doctor can be online from 6 a.m. I'll be
Yes,
chatting
that's
with him
what
from
I'm
6
saying.
a.m.
But
up
he
to
be waiting for
7:30.
us.
We don't come early. That is why at times we drive. And he is one person that he would like to stress on every point before he move on. He doesn't rush us through. So that 30 minutes would have done 2 slides or even 1 point. And we would delay. Look at the time that we said we were returning from break. 10. We came back 10:15. So it is we are at times we are to be blamed. So please, tomorrow we have all agreed. Tomorrow we are coming 7 in the morning. By 7:15, even if it is 10, we will start the class. Then we can finish on time. So that if you have any other programs, you can attend it. And as Victor said, it's a
Or even
weekend.
if it's 6 o'clock, right, then let's come because some of
Please,
us have
6
very
o'clock, I'll still be
important
sleeping.
programs tomorrow.
I'll be sleeping tomorrow morning, 6 o'clock.
Because me, for instance, tomorrow I'm on a program, very, very important one. So if we are to come, we should come early. Or since we have all agreed, let's come early so that we close early.
So we'll make it
very important too.
Start.
Julie, which center are you?
Mr. German.
I'm in
Julie, which
Sunyane.
center are you?
I'm in Sunyane. Yes.
All those around. Tomorrow, 7. No delay. We are not waiting.
7 and up and as a tumble.
I don't know, Pa.
We have been saying 7 and up and still you asking.
If anybody. Tomorrow by 7. I'm just going to
7 area.
You don't want to know.
7 area. Bye-bye. I have another class to attend. Bye-bye.
Mr. Bogaziman. Mr. Jiman. Hello, Mr. Jiman.
Anonya pastor woko. I'm somebody pastor man. Hey, intentional young question. Hey, asemo.
You could see I get into, don't worry.
I get.
Nti agasi the best atia kwa sorry.
Teacher. Teacher, good morning. And why you now say? What will you Nana? I'm just you're almost stressed so to talk.